Okay. All right, everyone ready? Great. All right, so hi everyone, I'm Daniel. Thank you all for coming and welcome to this workshop. Before we get going, I just wanted to say a few quick words of thanks to everyone that's making this workshop possible. First, of course, my co-organizer, Jennifer, who will be leading us through the second session of this workshop, has been just amazing to work with, and I'm really excited that she's soon going to be joining us here at Emory in January. We also get support through the Emory College of Arts and Sciences through their Theory and Modeling of Living Systems initiative, and also the National Science Foundation's Physics of Living Systems through the Student Research Network. I also wanted to especially thank Tiara Ward, who's done just an enormous amount of work behind the scenes getting logistics of this whole workshop series set up. On that note, just a quick plug for the next workshop in our series before the summer winds down. In August, we've got a two-part workshop on population genetics and microbial ecology lined up. That's going to be organized by Daniel Weissman. It'll have a slightly different format, split over 2 days with slightly longer talks planned. It should be great, and stay tuned to the Living Theory website for more details as they become available. All right, so why this workshop, right? There's been a tremendous growth in research at the interface between soft matter and biological systems. And so we asked our speakers a series of questions. We asked them, are all living systems also soft matter systems? Do the ideas of biological function or purpose require us to add fundamentally new ideas into the framework of how we do soft matter research? And does the often qualitatively different character of the data that we get out of biological systems require us to change the types of experiments we conduct or even the goals of our theoretical treatments? In short, we were asking them, where do they think the field is now and where is it going to be going? So that was a series of, I think, kind of deeply unfair, somewhat impossible questions to ask. And I'm just really excited to hear what they all are going to have to say to us today. On the format of this workshop, as we go through the schedule, each speaker will give about a 10-minute prepared talk on their view of the progress we've made and the challenges before us in this task. And then we'll have about 10 minutes of discussion. And we'll try to ask as many questions from the live streamed chat as we can. And I see that it's still currently disabled, but we should get that chat function up and running real quickly. All right, so enough of my talking. Our first speaker is staying at work late on a Friday from Germany, so no need to keep us all waiting. I'm really excited to introduce our first speaker, Chase, who's going to talk to us about dynamics and interactions of confined cell migration. So Chase, whenever you're ready, take it away.
All right, thanks, Daniel. Thanks so much, and thanks to you and Jen for organizing this wonderful event today. I'd like to start by just showing you a movie of a human breast cancer cells, a bunch of isolated cells on a 2D substrate. And despite this being a very boring environment for these cells, they still exhibit some spectacular migratory dynamics. So can soft matter physics actually help us understand such behavior? Well, cells are soft and highly deformable. And a lot is known actually about the rich mechanical properties of the cellular cytoskeleton. So this is an active material capable of generating stresses to deform the cell, capable of generating traction with an external substrate. So it's fair to argue that these soft matter properties are important, but cells can also sense. They can respond to external mechanical and chemical stimuli. And these sensory inputs, they can actually couple into complex biochemical networks, which regulate the cytoskeletal a motility machinery. So cells can not only sense, they may also be able to respond and adapt their properties and migratory behavior to an external environment. This is why I think it's important to study how the migratory dynamics of cells actually responds to structured environments and how it might respond to interactions with other cells. So let me show you a couple of examples. Here's a collection of neutrophils which are traversing narrow channels of an intravascular network. And a related example here in 3D is a leukocyte who migrates by actually squeezing through tight pores of a collagen network. So physiological environments like these are highly confining. And I think it's interesting to understand how a cell might actually be able of migrating through such a confining and restricting environment. But cells also interact with each other, right? So a classical example is this two-dimensional epithelial sheet. And another example here of which is currently a theme of a lot of focus are organoids, 3D clusters of cells. And understanding cell behavior in complex settings like these is a huge open challenge. The reason why this is so hard is because it's difficult to disentangle single cell behavior from how a cell might respond to its environment and how it might respond to interactions with other cells. So one way of tackling this is perhaps by creating artificial and highly controllable environments, such as microchannels and micropatterns, to help us study very systematically migratory dynamics. So today I'd like to introduce you to perhaps a minimal environment that we found to be really helpful to study the stochastic dynamics of confined cell migration. It's a micropattern coated with fibronectin, which promotes adhesion of the cell. So the cell really remains adhered and confined to this pattern. It's very dynamic though, right? It deforms quite dramatically also when it squeezes through this narrow constricting channel. So we'd like to see if we could understand this emergent migratory dynamics in confinement using some kind of simple underlying dynamical law. So we decided to track trajectories of the cell nuclei over time. So we get these stochastic cell trajectories, and we actually measured hundreds and hundreds of these trajectories. And then we used these data sets to actually infer an underlying equation of motion for confined cell migration. So this is a stochastic equation of motion, a Langevin equation, and it's a generalization of, for instance, the persistent random motion models that have been used to characterize freely migrating cells. So our task is now to use this big data set of trajectories to somehow infer properties like the amplitude of this Gaussian white noise here and the structure of this deterministic driving. So we arrive at a data-driven theory, which is largely encoded by how this deterministic driving depends on the position and velocity of the cell. So let me show you what we get. Here you can see what this driving looks like in a phase space of position and velocity. And there's an intricate nonlinear dynamics. And there's one feature I want to highlight here, which is best illustrated by looking at the deterministic flow in this phase space. So if we focus on this region over here, this corresponds to a point in the dynamics where the cell is moving onto this constriction. And we see that the cell is deterministically accelerating. And this is perhaps at odds with what you might expect if this constriction is some kind of repelling obstacle or some kind of energy barrier, right? So this deterministic acceleration or active squeezing is very robust. We see this for different cell types in different geometries as long as there is a constriction. So in this example here, there's no constriction. We see this active squeezing behavior vanishes. So this data-driven theory provides a dynamical systems description in terms of the nonlinear dynamics in phase space of different cells in different confining geometries. A major outstanding problem, though, is how the migratory dynamics of cells might respond to cell-cell interactions. So we started looking into this using a minimal cell collider. So here you can see two cells bumping into each other. They do this very frequently, and there's different characteristic collision behavior. Sometimes they reflect, sometimes they slide past each other, and occasionally you will even see the cells tailgating. again, we decided to use the nuclear trajectories of these cells as a low-dimensional representation of this interacting behavior. And we're actually going to compare two cell types side by side. They're both human breast tissue cells. One is a non-cancerous and one is a cancerous cell line. So we find that they have different kinds of collision behavior. So the non-cancerous cells predominantly reverse upon collision, while the cancerous cell line tends to slide past each other. Now, despite these differences in behavior and the large variability of this behavior, we'd still like to come up with some kind of coherent dynamical description. So again, we decided to use a data-driven approach. Now we're going to actually infer a two-body stochastic equation of motion for this interacting behavior. And in this case, we managed to reduce this to a simplified form where there is a single cell, one body term, which we inferred for these two cell lines. And they're actually almost identical for what we inferred for these cells when they're just moving by themselves in the pattern. Then we also measured the two body terms, which we actually split into a cohesive-like interaction term and a friction-like interaction term. And for the non-cancerous 10A cells, we find quite intuitively a repulsive interaction together with friction. For the 231 cancerous cell line, however, we find something a bit more surprising. So here we see an attractive interaction combined with anti-friction. And it's this kind of interaction dynamics which promotes this accelerated relative sliding behavior that we observe for these cancerous cells on long time scales. So this inferred dynamical description says something about the short time or instantaneous dynamics of these cells. And this framework can actually provide very accurate predictions for behavior on long time scales. Now, in throughout the cell biology literature, there's been lots of studies on different cell types, both in vitro and in vivo, showing many different kinds of collision behaviors. These have been termed things like contact inhibition of locomotion or contact following locomotion. So we wondered whether we could capture this diversity of behaviors using this dynamical framework. So what we did is we varied the amplitudes of this cohesive and this friction-like term, and we mapped out an interaction behavior space. So up here in this quadrant over here, we encountered the cancerous 231 cells in a regime where contact sliding dominates. In the opposite quadrant down here, we encountered the 10A cells where a contact inhibition of locomotion appears to dominate. All right, so as I'm about to wrap up, let me just say the main take-home message of today is we no longer have to guess the dynamics of cells. We no longer have to guess their interactions with other cells. We can actually infer this directly from data. This provides a stochastic equation of motion, which can make predictions for long-term behavior of these systems. And I think it's going to be a very interesting challenge to see how this would generalize by looking at other cell types and interactions between different cell types, and also in more complex geometries, both in 2 and 3D. A big outstanding problem is also to figure out how these kind of dynamics come about mechanistically, right? And here it's going to be important not only to use bottom-up models, but also specifically to look at how molecular perturbations of the cytoskeletal and adhesive machinery would control and affect this kind of behavior. I think it's also going to be interesting to see if we can expand these stochastic dynamical descriptions to also describe the full shape dynamics of these cells, and to investigate how the two-body interactions that we were able to infer here, whether they might actually be able to explain the large-scale dynamics of big clusters. Here, it might also be interesting to see if we can expand these inference approaches and directly infer the many-body dynamics of such a cell cluster. All right, so I'm out of time, but I just want to take this last moment to thank my group, which is actually moving now from Munich to Amsterdam. And I would like to mention a spectacular grad student in my group, David Bruckner, who's been driving this project from the very beginning in my group. And we've received a lot of fantastic experimental input from Alexandra, who's been doing experiments in Joachim's lab. All right, so thank you all for listening, and I'd be happy to take some questions.
All right, thanks, Chase, for really a wonderful talk. And I'm sure there's going to be a lot of great discussion stemming from it. I guess I'll ask the first question because I'm kind of quite interested. You know, there's beautiful work on thinking about how do we infer effective interactions in some of these systems. And you can imagine that this is useful not just in biology, but in all kinds of, you know, cases where you're observing the behavior of some dynamical system and inferring the force laws that come from it. And so I'm asking myself, what's different about some of these biological systems as opposed to any other type of system? And one could imagine that in these cellular systems, there's all kinds of co-regulation in how they sense the environment and how they interact with the environment. So for instance, how do we have a good instinct that, for instance, there's no coupling between some of the different, you know, single cell and cell terms that you've written down in this inference model?
All right, so those are actually a lot of interesting questions. So just to start with your last point, how do we know that there's no coupling? So we don't, right? And actually, it might be possible that you would have to describe these data with a more general formalism that would allow such direct coupling. What we did here is we actually started with a whole class of different models with varying complexity. You know, starting with the simplest model, you can actually infer the model from data and then make predictions with this model, which then end up failing, right? So then we decided to, you know, make these models more complex. So let me just give you a quick example. For instance, if you say, well, what if all the interactions are all going to be frictionless, right? We can still infer such a theory, but it makes very poor predictions. So what we did is we tried to just, you know, start from the simplest end of the spectrum and then slowly add complexity. And we just found that it is possible to come up with a predictive theory that has this simplified form. I don't know why these cells allow for such a decoupling. And you know, a priori, I had no, you know, clear expectations, just strong hopes.
Great, thanks. We have a question from one of our panelists, Josh.
Hey, Chase. So I have two short questions. Have you put one cancerous cell with another of the non-cancerous cell? And does that change the interaction? Presumably it does. And the second one is, have you studied how the interaction terms depend on the geometry of the chamber that you put them in?
All right, so first question. Yes, we would really love to do this, but we haven't been able to do this. Lab work is running a little slow these days, but I think this would be super interesting. There's a lot of interest throughout the cell migration community to understand the difference between homotypic and heterotypic interactions. We just focus here on the homotypic one because it's experimentally easy. You start with one cell, you wait until it divides, then you have two bumping into each other. There's no reason why we can't look at heterotypic interactions, so that's one of the next things on the list. I can tell you more about what happens when you change the geometry. So we did start looking at a few different geometries. For instance, a geometry where there's no constriction, just like a stripe, and then the cells keep bumping into each other, sliding past each other. Very interesting. If you infer a model for that case, the single cell term changes a lot. The interactive term remains almost unchanged. Again, we don't really understand why these things should decouple in such a simple way, but at least for the cases we looked at, they seem to.
Yeah, great. We've got a few questions from the chat. Ilya Nemanman asks, why is the model first order in time? I would have expected the cells to have internal states which would increase the order of the equations.
Right. So that's a great question. So I've actually always been kind of looking at, you know, how the description looks like in terms of the position variable, and in this case, you would actually pull the second order in time if we would look at, you know, acceleration and how that might depend on those things. We looked at overdamped equations of motion, so first order in position. They all seem to fail. Then we kind of understand why they fail, because cells have an internal state of polarization, which acts a little bit like to create an effective inertia. Then we decided to move to these second order or models in X or first order in V. Why they work as well as they seem to do, I mean, they're not perfect, but they have a lot of predictive power, I think is because we're mostly looking at dynamics on timescales of, which is basically set by our measurement interval. Roughly every 10 minutes, we take an image. I think that if we would start looking at shorter timescale dynamics, we should see evidence that there's like internal variables which are affecting our dynamics, which now are sort of all appearing in this white noise. So what I would expect to happen there is that we would no longer effectively be able to describe this as white noise. Whether we need to go to something that's even higher order than just first order in V, that I don't know. I mean, so far we've just been starting from the simple end and just seeing how far we can push this.
All right, great. We have another question from the panel. Cristina?
Yes, hi, Chase. Following up on Josh's question, even if you look at the interaction between same type cells, do you necessarily expect them to be reciprocal, meaning A does to B the same as B to A, at least in the context of effective interactions, which is what you are trying to calculate?
Right. So we don't have a reason a priori why to expect them to be reciprocal. What we've been assuming in our description is that there is an exchange symmetry, that somehow these particles interact with each other in the same way. So in that sense, it's reciprocal. And this allowed us to actually do this stochastic inference, because as you can imagine, doing stochastic inference with systems with several degrees of freedom becomes really demanding. So we've made the assumption that there is this symmetry, and we end up with a model that is quite predictive. But that's, I think there's no physical reason a priori why there should be this reciprocity. And it would be interesting to understand this a little bit better and to see if it might actually fail in some cases. I might expect that this is going to be especially interesting if we move to systems with even more than 2 cells. We've actually started looking at 3-cell systems and so on. And there, I think it's also going to be interesting to see whether, if you have 1 cell interacting with 2 cells simultaneously, whether the effect of those 2 interactions is an additive term, or whether we should really think of that as a inherently 3-body type interaction. So we're just exploring these ideas, but since this is an active system, there's no, and this is a very effective description, there's no reason a priori why to expect reciprocity.
Thank you.
Yeah, somewhat following up on that question of how this is an active system, from the chat, Ricard Zenon asks if you can interpret the velocity-dependent forces in this model, the friction and the anti-friction, in terms of active cell-cell interaction forces.
Right. Yes, we're thinking very deeply about this. So one big difference between these 2 different cell lines is how many EK adherents are expressed. So this is a transmembrane protein. The 10A cells have lots of these proteins, so they're pretty sticky. So from that point of view, we were kind of expecting something like a friction-like interaction. The thing we can't really explain very easily so far is where this anti-friction behavior comes from. We have some insight, which we actually obtained from the single cell behavior. There we saw that whenever the cell needs to kind of squeeze, it starts accelerating, right? So it is possible that the basic behavior of these migrating cells, that whenever they're kind of very restricted, that they actually start accelerating. This we're trying to understand with a very simple active particle model, which has like an internal state of polarization, and whether that would naturally lead to these behaviors. But it's not obvious how this anti-friction would come about, in my opinion.
All right, thanks very much. Well, that was a kind of fantastic talk and a great set of questions. Unfortunately, we are already out of time to take more, but so thank you, Chase, again.
Thanks.
Yeah, and up next, we have Mike Murrell, who's going to tell us about capillarity and wetting in active systems. So Mike, whenever you're ready, take it away.
Okay, can you see? All right. Okay, so in this session, we want to understand what biological principle can be learned by modeling biological processes using interfacial thermodynamics, and in particular, models of capillarity and wetting. So wetting, of course, describes the process by which a fluid will create an interface, like a droplet spreading onto a rigid surface. Capillary forces are going to arise from the interfacial energies between the solid, liquid, and vapor. These energies will then determine the shape of the droplet and are going to determine the dynamics of its motion. So of course, in biology, we're really interested in understanding changes in cell shape, and we're interested in understanding the dynamics of their motion. So everything from how a single cell changes its shape as it spreads onto a surface, to the collective motions of epithelial sheets, or the movement of clusters in development. So the basic idea is, can we use these kind of generic models to understand something about complex biological behaviors? And so essentially, we want to use models of wetting for a variety of different model systems to be able to understand mechanisms for dissipation, in particular active dissipation. We're interested in understanding if there are universal relationships, there are common ways in which systems that vary dissipate energy similarly. Are there ways in which they do this similarly across scale? And also, we're really interested in understanding when these thermodynamic models break down. Some of these passive models are, of course, used for passive processes, but can actually well approximate active processes. So when do we need active models, and when can we get away with passive ones? So as I mentioned, just very briefly, of course, wetting involves the creation of this interface between a droplet and a surface. It's determined by intermolecular forces between the fluid and the chemistry of the surface. This is what yields an interfacial energy, and there are three interfaces. These three interfacial energies act as tensions, which at equilibrium balance to describe an equilibrium contact angle. Now, when non-equilibrium, then there's a net force, a capillary force, which is going to drive motion. So we intend to derive analogies to these capillary forces to the motion of living material. So to understand the dynamics of wetting, what we do is quite simply, we balance the gain in energy that occurs during spreading with the dissipation of energy. So the gain in energy occurs because the droplet wants to maximize its adhesive interface, gain more adhesive energy to minimize its free energy. But as the droplet spreads out, there'll be a flow of liquid. This can be described by Navier-Stokes, of which the only term which remains are the viscous stresses. The overall dissipation rate then will be a factor of the viscosity of the fluid, the strain rate, and the volume over which energy is dissipated. So in balancing these terms, we can get a relationship between the spread area and the scaling with time. So for a liquid droplet, we see the area goes as time squared, and the rate is determined by the balance between the adhesion energy and the viscosity. So we can use this to try and explain a variety of biological behaviors, and I'll give a couple of examples, one quite past, one more present, and one we're continually working on. Years ago, it was shown that the dynamics that describe how different cells can adhere and spread onto the surface are very similar. So whether they're red blood cells or fibroblasts, basically they would create an adhesive interface, A, that scales linearly with time. This was called the universal dynamics of cell spreading. Now, we hypothesized that there was a dissipative mechanism which was kind of dominating this behavior, giving it this universal scaling. So what we did was we created a biomimetic cell where we could put the actin cytoskeleton inside of a liposome in this kind of cortical shell. If now, like a droplet, we drop this onto a surface, it adheres and spreads. For an empty vesicle that does not contain actin, dissipation occurs throughout a volume, so that would be R cubed. The strain rate would be 1 over R dr dt, and we would recover the same scaling relationship that we got for a liquid droplet, with area going as time squared. However, for a cortical shell, we end up getting area going linearly with time only when we say the dissipation happens within a disk with a constant thickness W. In that case, we get area going linearly with time. And so with this very simple scaling relationship, we were able to identify which of these different dissipative mechanisms is responsible for spreading behavior. We can also take an example from a more active system. So here we have an aggregate of cells that is intended to model a tumor. And cells, when interfaced with an adhesive environment, will spread by migrating out of the aggregate. And so this is an active process where cells will reach the bottom surface and they'll migrate radially outwards from the center. But as you can see, this looks very much like the spreading of a droplet. So now you have a film emerging very similar to that of a precursor film in a liquid droplet, but this occurs through migration. So while dissipation occurs again throughout a volume like R cubed, the dissipation actually happens basically purely at the surface. And so there's a constant in the strain rate. And that gives us a scaling relation as T to the 2/3, which is exactly what is measured experimentally. And so in this case, we were able to show that we get this coefficient because we have something called a permeation width, in which cells will specifically and only move into the contact line as they spread outwards. Now, in this active case, we know that cells can be very mechanoresponsive. So in not all cases do they actually spread as droplets. So now if we have an elastic surface, we place cells onto a polyacrylamide hydrogel, which can be made very compliant. When the surface is very compliant, the aggregates do not spread, but seem to kind of diffuse across the surface. Interestingly, at somewhat of an intermediate stiffness, these aggregates will wet the surface, they'll emerge a monolayer, but then over time, the monolayer will retract, become unstable, and the aggregate will de-wet and migrate away. And so we were curious as to what could be responsible for the de-wetting. One can represent the motion of a droplet on a surface through differences in adhesion through capillary forces or elasticity. So it's been suggested that cells can actually modify the adhesiveness of the surface that they're on. And so perhaps the cells are remodeling the ECM front to back. So that would yield a different interfacial energy on one side versus the other, which will create a net force, which has to, of course, balance the frictional force. Again, by balancing the kind of adhesive power with the dissipation rate, one can isolate the net velocity, which will be in terms of the difference front and back of the adhesive energy. This gives a simple relationship which requires no external activity. Alternatively, one could propose an active model and modify these passive wetting models with activity, which are inclusive of the active stresses that are generated, which we can see by traction force microscopy. Typically, stresses are predominantly located on the periphery of the film as the droplet is spreading. And in addition, there's polarity in the organization of the cells in their shape, with cells being more polarized on the periphery and less so in the middle. So we can incorporate the stress field, the velocity field, and the polarity field. So we could decompose the traction into stresses that come from the polarity and also retain a friction term. The polarity is going to be largest at the periphery and decay towards the center. So it'll follow a diffusive profile for which we can take a mean, which will give us a kind of a linear profile. And if we take the average of that, again, we get a relationship for the motion of the aggregate, which actually has basically the same form as the passive model. So instead of the forces being driven by a difference in adhesive energy, and so a difference in capillary forces, we have the forces arising from a difference in polarity with essentially exactly the same form. So the question is, when can we use these passive models, and when are they active? When should they be modified with activity? As you saw, even for a case that was clearly active, the motion of the aggregate, we could suitably represent that with this kind of basic thermodynamic model, and that it very much resembled the activity. But not all cases can this be uniformly applicable. So in our group, we study collective migration of epithelial sheets. Here you see a movie of actin within an epithelial monolayer. We wounded it in the center that creates this hole, and cells will migrate to close the wound, and they'll generate these forces while doing so. What we find is that over time, the strain energy decreases, and so and it does so at essentially a constant rate. And so we find, like these passive systems, that we have this kind of constant dissipation rate. However, there are embedded levels of activity which give it this constant rate. So for example, at the periphery of the wound, cells will choose one of two different morphologies in migrating. They'll either use this kind of lamellapodial migration, which happens to be quite fast, but generates very low forces, or they'll use this more purse string-like motion, in which motion is slow, but generates really high forces. Now, as the wound closes and maintaining this constant dissipation rate, however, it'll actually switch between these two different modes, and it will switch in accordance to keep this power, this dissipation rate constant. And so despite the fact that at the kind of tissue scale, the mesoscopic scale, indeed we have a constant dissipation rate, activity is used to choose which mechanisms the monolayer will use in order to be able to close. And thereby activity is what's generating what at the surface looks to be a passive behavior. So our assumptions for being able to use these models is clearly we need to be in a regime in which we can equate these two different types of adhesion, these two different types of energies per unit time, this gain in adhesion and the dissipation rate. But more specifically, it matters that these adhesion energies and viscosities, be non-constant. In these cases, we are literally exchanging cases where adhesion is high versus when adhesion is low. And we do that to be able to maintain this constant dissipation rate. So with that, I think I'll end there and take any questions you have.
All right, thanks, Mike, again for really an excellent talk. And I really liked kind of the emphasis of a lot of what you were asking, when saying for some of these passive thermodynamic descriptions of what's going on and asking, is that all there is? And it also kind of gets to the heart of, what do we mean when we say we're modeling the behavior of a system with a particular reduced dimensionality model? So one of the things that I wanted to ask you about before turning it over to the rest of the panel, for instance, in these beautiful cell spreading experiments, both in the case where the passive and the active models fit, even still, the structure of the interface, kind of as you allude to with these wound closing experiments also, the structure of the interface looks so different than normal wetting of passive molecular fluids, for instance. So I wanted to ask if there's kind of something in the structure of these interfacial fluctuations that kind of point towards inadequacies in the type of modeling that we're doing to discuss these systems.
Yeah, there is. So, for example, in this case with wound healing, the choice of motility mechanism that your neighbor chooses influences your choice. So there's actually, it's not a random process. tension accumulated, for example, by a purse string with neighboring cells makes them more likely to create purse strings. So I think in these cases, we would, because of these kind of like interdependences and nonlinearities, we end up with, energies that cannot be described by these passive thermodynamic models. So in that sense, the structure of the interface is exactly what's determining, the balance of these dissipation rates. It really comes from how the choice of motility mechanisms influences your neighbor's choice of motility mechanism.
Yeah, thanks very much. Okay, so next we have a question from one of the other panelists, Eleni.
So hi, and thank you for the great talk. I became very intrigued when you showed these two types of behaviors that seem quite distinct. So my first question is this really a very distinct behavior where you can say, okay, this is by model, or is there a continuum? I didn't get that very well from your talk, but what was also quite interesting is that there is feedback by what collectively your system is doing, and what the cells do in order to implement maybe the optimal behavior for the system. So I was wondering if you can talk a little bit more about that.
Yeah, so in terms of the continuity of shape and motile mechanism, they're actually pretty separate. So they use different assembly factors for the cytoskeleton, F-actin nucleating proteins. So this behavior on the left is a formation of something called a Lamellapodia, which is a large branch structure which assembles quickly and drives fast motion. This case involves the assembly of bundled structures, which assemble myosin and contract and generate large forces. So there's actually really not much of a continuum between them. They are pretty distinct. And yeah, you're right that actually it would appear that the dissipation rate is being set by the tissue, because the choice of cell architecture individually can be either. And so overall, the tissue is basically determining what the individuals should be doing and setting that rate. Yeah, exactly.
All right, great. So we have a few questions from the chat. So it looks like first, Top on Goal says, great talk, and asks if there's a reason to assume that the traction arising from the polarity is linear.
It doesn't need to be. The polarity can be measured, and the polarity is measured to be roughly linear, at least for small aggregates. As the monolayer increases and depletes the aggregate, it actually does become pretty nonlinear. So we basically just make that approximation.
Mm-hmm. Great. Okay, Suraj Shankar asks, is there something deeper that one can take away with the constancy of dissipation? Is there something that may generalize or not? And do you understand why that is beneficial biologically?
So great question. That's essentially the goal of our group, is to try and understand, what principles are driving motion. Our, interests in studying these non-equilibrium systems and trying to estimate the dissipation of energy is to understand what is consistent with what we think the goals of the biological system are. So for example, with the closure of a wound, we assumed that the system was optimized maybe for closure speed. But actually, if you choose all of these lamellapodial motions, you would actually close fastest. Instead, you use a mixture. Or one could assume maybe that you want to have a really strong closure, in which case you would have all of these on the right-hand side. But again, you don't. You have a mixture. So the mixture seems to maintain kind of a constant dissipation rate. So I really have no idea why the system would necessarily want a constant dissipation rate. We basically see it as a bound on the energy produced, because the dissipation will be limited by the consumption of chemical energy, but also by practical features like the remodeling of the cytoskeleton. So it could just reflect a bound on what the machine is able to do.
Mm-hmm. Yeah, and I think that gets to exactly some of the questions that are still popping up in chat about the distinction between kind of these as an active system versus these as a living system. So a lot of great additional questions showing up in the chat. Unfortunately, we don't have time to address all of them, although maybe Michael will pop into the chat and answer some of them himself. But thank you for a fantastic talk. And up next, we have Cristina Marchetti, who's going to talk to us about symmetry breaking, geometry, and topology in biological systems. So Cristina, whenever you're ready.
I didn't unmute myself. Okay. Thank you, Daniel and Jennifer, for organizing this. Geometry and topological frustration have played an important role in soft matter, and have certainly provided us with opportunities for the design of new materials with tailored functionalities. So what I hope to do today is to show you that the same ideas can serve as well in biology for connecting patterns of molecular level signaling to organization and function at the tissue scale. Yes. Okay. So in soft matter, we know that ordered states of matter, such as a ferromagnet or a pneumatic liquid crystal, are interrupted by topological defects, singularities of the ordered parameter field that cannot be eliminated by smooth deformations, and can be generated, for instance, when you quench a system from the disordered into the ordered state. In a system with a polar order, as the one here on the left, the lowest order topological defects, the ones that are energetically more favorable, are vortex, asters, or what is called cross-air, defects that have topological charge +1, where the topological charge, or winding number, is defined as the angle through which, as you see down here, the order parameter rotates when you circle the defect with a closed contour. In pneumatic liquid crystal systems where the elongated molecules are aligned, but have no head and tail, there is no preferred direction. The defects that have the lowest energies are disclinations, which have a charge +1F or -1F. The +1F is the distinctive polar-like structure, sort of like a comment, and the -1F is this trifold symmetry. The important thing in equilibrium is that we understand how to map the statistical physics of defects onto that of Coulomb charges, and opposite sign defects attract just like charges and tend to annihilate, so that the system eventually reaches an ordered state. In active matter, in active systems, the main difference is that topological defects become themselves motile entities. In active pneumatics, like these two-dimensional suspension of microtubule bundles at an oil-water interface that I'm sure we have all seen before from the lab of Zwalimer Dogic, you see the spontaneous generation of plus and -1F disclinations, and the +1F, the ones that look like a comet, are actually behaving themselves as self-propelled particles. In polar active fluids, this is from the lab of Denis Barthelot in Lyon, and it's essentially a system of active colloids, the so-called Quincke rotors. And what happens is that in this case, the system is polar, so the topological defects are vortex or aster-like, and these asters become actually self-rotating spirals. So again, the main point is that they become motile entities. Well, what about in biology? Do defects actually play a role in biology? What I'd like to show you as an example is some beautiful recent work from the lab of Kinneret Keren at Technion on Hydra, a freshwater organism that can regenerate its entire body from a tissue fragment. The body of Hydra is kind of like a tube with a foot at one end and a head at the other end. And it shows a lot of large-scale nematic alignment of atomizing fibers, essentially. And when Hydra regenerates itself, starting from a fragment, what Kinneret has shown is that, first of all, the fragment becomes essentially a spheroid. And on a spherical surface, nematic order cannot, of course, be accommodated exactly. So you require topological defects, either 4+1F, or like in a structure of a baseball, or 2+1, as shown here. And what happens in Hydra as it regenerates is that first there are +1F defects generated, which are motile, and recombine into +1 defect. And the +1 defects are actually the places where the head and the foot of the animal are generated. So we could say that in this case, topological defects in the nematic texture serve as organizing centers of morphogenetic effects. So they do serve a biological function. There are other examples of topological defects that serve a function in biology. They have been shown to, again, +1F disclinations in nematic order of cell orientation have been shown to serve as centers of extrusion and cell death in epithelia, and of mound formation in neural stem cells. And the movie is actually from our next speaker, and I'm sure he'll tell you more about it. It's a myxobacteria, and it shows that topological defects are associated with either holes for the -1F or multi-layering, which will lead eventually to the formation of 3D structure for the +1F. So with this motivation, as well as motivated by the fact that in equilibrium, again, we know that we have this mapping of defects onto a Coulomb gas, which is behind the physics of a lot of phase transition into dimension driven by Kosterlitz-Thouless defect unbinding. So with this motivation, we have embarked on an attempt to formulate essentially a theory of active defects, especially in active nematic liquid crystals, as quasi-particles. And what we have been able to show is that we can actually map the dynamics of defects, the plus and -1F disclinations, onto that of a mixture of self-propelled particles, the +1F, and passive particles, the -1F, interacting with Coulomb forces, but also with aligning torque. One important outcome of this work is that in active systems, an important degrees of freedom of the defects, in particularly of the +1F, although not only of the +1F, is their polarity. So we developed equation of motion for defects position and orientation, and some more recent work, including multi-defect dynamics, has also revealed the non-reciprocal nature of these interactions, although I will not say much about that today. So I just want to highlight a few of the outcome of these defect dynamics that I think are important in the context of biology. So first of all, as I already mentioned, the active defects, the +1F defects become a motile particle, and we can understand easily and in fact calculate the defect motility as arising from the flow that the distortion of the nematic order due to the defect itself generates. The distortion of order generates this vertical type flow shown here in blue. The flow is non-zero at the defect core, and the defect itself sort of behaves like a colloidal particle in the flow and moves with the flow it itself generates. Due to this symmetry, the velocity at the core of the -1F is 0, and the defect is not a self-propelled particle. But an important thing is that the direction of motion of the +1F depends on the type of active stresses at play in the system. It's towards the head if the stresses are extensile, and towards the tail if they are contractile. And what that means is that defect motion can actually be used to identify the nature of the active forces, distinguish extensile from contractile forces. The dynamics of defects in this sort of mean field approximation, where we focus on the motion of one defect in the field of other defects, is distinguished from equilibrium. Again, you have interactions which are Coulomb-like, but the key point is that the +1F defects defect has this propulsive force here and experience active torques. It sort of behaves like a dipole in the electric field due to the elastic deformation, the Coulomb forces of all other defects. And these active torques are important in the sense that they, first of all, stabilize bound states of 2+1F defects, which have a different structure depending on whether the system is extensile or contractile. And in particular, for extensile system, the bound state from a distance looks like a +1F aster, which is precisely the type of structure that occurs in Hydra, where 2+1F sort of apparently seemingly recombine to actually form the aster that eventually seeds the formation of the foot of Hydra. And indeed, in Hydra, the motion of the +1F defects reveals the extensile nature of the forces. Another effect of these active torques is that first of all, our equations have allowed us to construct a phase diagram for defects in the plane of activity and nematic elasticity, both quantities that can be tuned in experiments. Phase diagram itself is probably more relevant to in vitro system. It shows that defects, activity unbinds defects, leading to this state of active turbulence seen in the microtubule nematics that resemble sort of cytoplasmic flows inside cells. But most interesting, you find that at even higher activity, defect-defect interaction can actually lead to an emergent state where defects themselves order into a flocking state that has been seen in simulations, so far not in experiments. And there are actually open questions on whether, on the precise nature of the order, of the possible order of defects in this kind of systems. And then finally, activity gradients generate again elastic deformation of the nematic texture. And one can show that they can act effectively like an electric field to actually direct and confine defects. Again, the effects behave like a dipole in the field now generated by activity gradients. And so one can actually wonder whether nature might be able, and this has been seen in experiments in atomizing suspension. The top image is a cartoon from calculations we have done, and the bottom are experiments from the group of Margaret Gardell at the University of Chicago. But one may wonder whether nature may actually use in, say, development and cell rearrangement in tissue may use activity gradients to actually direct defect motion and associated cellular flows. So with that, I would like to conclude to kind of try to summarize what I've been trying to say and highlight a few important, I think, open questions in this area. So there is evidence in biology that defects may act as organizing centers of tissue morphogenesis, and the property of defects in active systems are quite relevant for these. First of all, the motile nature of defects that are driven by flows and then themselves generate cellular flows. Defects can be used to identify the nature of the active forces and make themselves organized in order structures, and actually order defect structures have been seen in bacterial walls. There are lots of things here to be explored, and an important, of course, we need to explore the interplay between geometry and topology. The Hydra, after all, is on a curved deformable surface. What role does that play in directing the motion of the defects? An important question is actually how do active forcing and tissue response manipulate cell organization? What is the feedback between structure, mechanics, and biological signaling? And the role of non-equilibrium noise and reciprocal interactions. In general, the kind of question one is asking here, the relevant biological questions, how do we connect biochemical signaling at the molecular scale to mechanics of function at the tissue and organ scale? And the route that we are taking, inspired by soft matter, is to essentially focus on describing molecular processes such as the supracellular actin organization in terms of effective coarse-grained fields, and then look at the dynamics of these fields, describe their dynamics in terms of collective excitation, such as in this case, the defects, and look at their roles in driving tissue morphogenesis. And if somebody asks me, I can say a few words about a different type of topology, something called topological protection, that has been playing a big role in condensed matter physics, and whether that could possibly play a role in biology. But with that, I will stop here, and maybe I'll just put up for a minute the image of my group in Santa Barbara, and thank the people with whom they've done this work, which is really mostly due to a great former student, Suraj Shankar, now at Harvard, who I guess he's listening since he asked the question a minute ago, but also in collaboration with Sriram Ramaswamy, Mark Bowick, and most recently with Boris Schreman. So thank you. I hope there are some questions. I'd like to go back to this slide here.
All right, thanks, Cristina, very much. Fantastic talk. And I want to follow up a little bit on this kind of question about the role of topology in active matter versus the role of topology in living matter. Of course, in soft matter systems, we're so used to thinking of topology as this wonderful kind of robust organizational principle. And I wonder if you think that the same will typically be true in living systems. I guess I'm particularly thinking, for instance, about the role of topological defects in some of these epithelial monolayers where defects are, potential birth-death sites or extrusion sites for the cells. But the fact that the defects are being described kind of at the same scale as the units themselves, I wonder if you think that will lead to a new way that topology actually enters the problem.
That's a very nice question. So first of all, I think it's clear that systems like epithelia are highly disordered systems. And we are talking about defects in order system, but in a sense, the order system itself almost does not exist because certainly we haven't seen cell layers that exhibit kind of perfect order. But I still think the idea of trying to identify, so what I have in mind here is that if we can identify sort of what we call them morphogenetic fields, like in the Hydra, it could be again the distribution of supracellular atomyosin, and describe this as a coarse-grained level. We, in some sense, in that case, we're not thinking about individual cells at all. We are just looking at a field that describes processes actually at the smaller scale, at the molecular scale, and we describe it at the coarse-grained level. If we can identify these kind of characteristic excitations, which are the defects of looking at the dynamics, that seems to be a very useful approach. And in the context of cells, I think Josh might say a little bit more about this, but calculations have shown that although indeed the defect core, which is the size of these particles, may be actually comparable to the size of the cells themselves, still the approach seems to be a useful one for understanding the dynamics of this system. I think there are other examples in soft matter where we actually use a continuum model to look at scales which are the scale of the particle. For instance, we use continuum model to describe neutron scattering in fluid, and it works remarkably well. And so it's kind of the same philosophy here, I think.
Right. Thanks very much. Briefly, we have a lot of people in the chat asking you to say a few words about topological protection in biology.
Okay. Well, there is nothing to say about topological protection in biology. That's the point. So, first of all, in some sense, topological protection comes from the notion that there can be topological defects in reciprocal space, meaning singular texture in the band structure of a material that affect the way waves propagate through the material and lead to things like topologically protected edge state that can be robust against disorder and backscattering. And of course, these are the kind of phenomena that have been studied for quite some time in quantum systems. They are behind the quantum Hall effect. And then for quite a number of years in soft matter system in the context of mechanical, photonic, and acoustic materials. Now, most recently, we have seen that topological protection can play a role in active matter, and I distinguish that from biological systems in the sense that there have been work by us and by the group of Vincenzo Vitelli showing that in suitably confined, let me say, polar active fluids, you can actually have unidirectionally propagating sound modes on the edge of the system. And this is between microfluidic networks and at the equator of a sphere, for a polar fluid confined to a sphere. Now, I don't think, as far as I know, to my knowledge, there is any example of this kind of phenomena in a biological system. And one wonders whether nature could possibly use topological protection for a robust transmission of information, because after all, these modes are robust, as I said again, against disorder, against backscattering. And it's certainly something that I would like to see explored and worth exploring a little bit more. So I just wanted to put a plug for this, but I don't think I know of any actual example in biology.
Yeah, exactly. Well, thank you again. Yeah, I agree. It's a fascinating question whether biology can exploit this mode of information transmission, as you say. All right. Well, thank you again, Cristina, for that excellent talk. Finishing up the session, we have one more talk by Josh Shaevitz, who will share some thoughts on active and passive dewetting in biological systems from molecules to populations. So, Josh, whenever you're ready.
Josh, I think you might be muted.
Did that fix everything? Hello?
Yep, we're good.
Excellent. All right. Well, thank you for the invitation to speak today. I don't think I realized I was studying something called soft matter until someone told me that at some point. But I do study a number of different biological systems. And today I want to tell you a bit about the interaction between liquid-like systems and solid-like systems, both at the molecular scale and the population scale. And we'll see aspects of both passive and active things that have been mentioned by Mike already, things that have been mentioned by Cristina just now in terms of active liquid crystals and things like that. So to start off, I want to talk about proteins. Proteins obviously can form solids. So a microtubule is essentially a solid form, a solid aggregated form of proteins. But as you also know, recently it's been discovered that proteins can form liquid-like states in which there is an aggregated state, but the ordering of the proteins are able to move around. It has viscous properties, it has surface tension properties. This is often called liquid-liquid phase separation. And what we've been seeing very recently is that there's a number of different systems in which liquid protein states interact with solid proteins. In particular, there's a number of liquid protein droplets that interact with microtubules themselves and sort of wet and then also de-wet onto microtubules. So here we see a number of different protein systems in which the liquid protein state has wetted along a microtubule and in some cases de-wetted. Okay. So what I want to tell you about is that this process actually is fundamental to the way almost every microtubule in your body is made. And trust me, there are a ton of microtubules in your body. Many of them are made in the mitotic spindle. So we're going to use that as an example, but this is going to apply to many other kinds of microtubules that you have in your system. So in the mitotic spindle, many microtubules are born at the poles of the spindle in what's called the centrosome. And the centrosome is decorated with a molecule called gamma Turk, which nucleates microtubules. But the vast majority of microtubules are not originated at the centrosome. The vast majority of them are born as branches off of the side of existing microtubules, so that you get geometric growth of the number of microtubules in the spindle. And so that happens using this nucleating protein, but that nucleating protein needs a bunch of other accessory factors to localize to the microtubule and do its job. And those accessory factors correspond to the things on this list. There's quite a few of them, Augmin, X-maps, things like this. But the major player, the one that localizes it first and decides where the new microtubule branches are going to be, is called TPX2. And TPX2 is in its active form near the chromosomes in a mitotic spindle. Basically, TPX2 is everywhere in the cell, but it's in an inactive form. And there's a gradient of something called RAN-GTP that's released from the chromosomes, and that gradient activates the TPX2. So you have activated TPX2 in the middle of the cell. And what you're going to see here in this movie, the chromosomes are here labeled sort of in blue. You're going to see red microtubules that are growing. This microtubule happened to get stuck. It ran into a chromosome, and you're going to see there's a ton of branching that happens. And in fact, now a gigantic amount of branching that happens there. And so that's where all the microtubules essentially are being born inside of the cell. And this TPX2 is not a protein that just binds a microtubule and finds its buddy. It's a protein that actually forms a liquid-like state. That liquid-like state wets microtubules, but at physiological concentrations, this wetting is too small to be seen. It's tens of nanometers. You can't see it with fluorescent microscopy. So we've been using atomic force microscopy. Here's an atomic force microscopy image. You'll see a movie. We flow in TPX2. It immediately coats the microtubule, but then you might see that the density kind of breaks up. And if you look at the color map here over time, what you see is the density starts out, there's nothing there. It jumps by about 15 nanometers. That 15 nanometers then goes from smooth to corrugated. If you look at a scan across the microtubule, you see that it's initially flat, that's the blue line. And then it breaks up into essentially a sinusoidal pattern that has a very dominant wavelength. So what we have here is an example of the Rayleigh plateau instability. The instability that happens when I have a fluid film in a tube that makes it want to break up into droplets. It's a surface tension dominated phenomena. It's what makes the water coming out of your faucet break up into drops if you make the flow rate go down. And in fact, the spacing, the wavelength that you get is a geometric quantity. So if you write down the theory at low Reynolds number for the Rayleigh plateau instability, what you find is that you can predict the droplet spacing as a function of the film thickness. The microtubule thickness is fixed, so the film thickness with no free parameters. And indeed, we do pretty well. We can control the film thickness. We then measure the droplet spacing, and we do pretty well there. And in fact, these droplets are where all the microtubules are born. If you want to know more about this, I'll have a link at the end of the talk to a paper that describes it. So this is an example where I wet a microtubule, I then de-wet it, and that de-wetting process produces localized droplets that are then going to be essentially the birthplace of all the microtubules in your cell. So that's a passive de-wetting. It's essentially classical hydrodynamics, the way you may have learned it in undergraduate. Now I want to talk about an active de-wetting. This is something that Cristina was referring to. We have a single cell in the upper left here of Myxococcus xanthus. It is a bacterium, it's elongated as a rod, it's kind of flexible, and it moves on surfaces. It doesn't swim. And when you put hundreds of thousands of these or millions of these together, what you get is a large film that coats, in this case, a flat surface. And what you see is that the film de-wets into small droplets, similar to the kinds of de-wetting that Mike was describing, except that now this is driven purely by active processes. And in fact, as I'm going to show you, it's not driven by the line tension at the contact angle. It's driven by the residual stresses you get at topological defects in the pneumatic liquid crystal. So if you look at the early stage of this, when it's very wetted into a monolayer, and you zoom in, what I hope you can see in the zoom resolution is that you have lots of single cells, and they're all pneumatically aligned in the kinds of liquid crystal or pneumatic systems that Cristina was telling you about. And now you can watch these move in a movie, and you see that the population starts as a 2D layer. It breaks up. There are holes that form that rip open in the layer, and there are second layers that form on top of the first layer. There are then third layers and fourth layers, etc. And these layerings are what's going to give rise to the droplet formation, to the de-wetting of the population off of the surface. So to give you something that looks a little bit more like what Cristina was showing here, we've just drawn the streamlines from the pneumatic order. You can see that there are topological defects, there, and there, many of them if you look around. And if you look carefully, you can see the topological defects are associated with the formation of holes, which are the dark brown things, and the formation of second layers, which are the white regions here. Okay, so this is a movie that actually Cristina showed you where we plot the negative 1/2 defects in blue, the positive 1/2 defects in red. And you can see that when you get the formation of a new layer, that happens at the particular place of a positive 1/2 defect. And if you get the formation of a hole, that happens at the place of a negative 1/2 defect. Or when the hole closes, it closes into a 1/2 charge defect. We can then write down a theory for this active liquid crystal. It turns out mixo go sort of back and forth, and so they act like an extensive liquid crystal. And then you can compare the kinds of flow fields and stresses that we find in the experiments to what you get from the theory, and we get quite good comparisons. Now, one thing that Ilya has been asking in the chat is what separates active from biological. And here's, I think, an example where I can say not only is this important for the function, but it's something that's really interesting biologically in that the cells control the activity. And so they can decide what kind of behavior they're going to do as a population. So Myxococcus xanthus sometimes likes to be wetted on a surface. And it makes a collective decision to try to de-wet into droplets. And in fact, these droplets give rise to what are called fruiting bodies. These are macroscopic. So this is like a picture from a cell phone camera. They're macroscopic objects full of millions of cells that then sporulate, and it forms sort of a protective system. And so they don't want to de-wet all the time. They want to change their behavior to de-wet when, for example, they're starving. And so you can experimentally form a phase diagram of this. You can do a little bit of theory work, which we did with Cristina. And you see that when the cells are happy, they set the parameters of their motility, which give rise to this inverse Peleg number, into the wetted regime of the phase diagram so that the population stays wetted. And when they're starving, they change their motility, which lowers the inverse Peleg number. They drop into this regime of the phase diagram that causes the system to de-wet. So not only is it active, but the biological part, at least partly, comes in the fact that the cells themselves and the population as a whole are sensing their environment, altering the activity of the population in order to affect these kinds of phase changes. So that was a very quick thing about a lot of different work. If you're interested in the microtubule work, that's a collaboration with Sabina Petri and Howard Stone at Princeton and was done by 2 students, Bernardo and Sagar. The Mixo work was a compilation, a very brief compilation of a couple different papers. You can read the references there, and was a collaboration with a number of groups, including Cristina and Ned Wingreen and others. And with that, I will stop and take questions. Thank you very much.
All right, thanks very much, Josh, for fantastic talk and a beautiful set of experiments and movies. I'm going to combine half a question of mine with a question in the chat from Siva Surender Chandran, who's asking why the de-wetted holes in those experiments are circular, and also, do they have a characteristic length scale in these systems? So
that's an interesting point. The de-wetted holes are not actually circular. There is a bit of a line tension, but because there's a couple of things. Because of the nematic, the elongation of the cells, they tend to look a little bit elongated. The other interesting thing about holes is they can absorb lots of charge because they're at a boundary of the liquid crystal. And so you can rip open a hole without a defect that looks like that. I don't know if you can see my video, but if you can, and then you can close it back like that. Or if you rip open a hole that starts as a -1/2 defect, then it looks like a triangle. That's a little bit harder to close. Then you can absorb another -1/2 defect, and then you look like a larger structure. And so the symmetry has to do largely with the charge of the edge state of the hole.
Great, thanks. Thanks very much. Another question that I had actually, in this last section, you told us about kind of the living matter aspect of this, where the cells are able to regulate the term that you're calling the activity in this model of active liquid crystals. But is there evidence that these biological systems could also be regulating all of the other effective parameters in this model? And if so, I wonder what your comments on kind of what it means to model them in this way is.
So there are a couple other parameters that are not in the model. One has to do with adhesion to the surface in certain ways. The other one has to do with, the surface tension with the outside, or something about the line tensions. Those do not appear in Mixo to be under much control by the cells actively, in the sense that when we try to measure things, those parameters don't seem to change depending on what you've done to the cells and stuff like that. But it's certainly possible, and definitely in other systems, changing substrate adhesion and cell-cell adhesion is a large part of the way the active processes happen, in eukaryotic cell sheets and things like that. And so those definitely can't happen.
Great, thank you. Let's see, also from chat, Paul Baconier says, thank you for the great talk, and asks, to what extent can the bacterial film dewetting and clustering be interpreted in terms of motility-induced phase separation, if it can?
So it's kind of qualitatively, it's quite different. The traditional motility-induced phase mechanism, or phase separation mechanism, is one of jamming, in which cells come together, they collide, they're jammed, and they can't move until one of them happens to point in a way that allows them to move away. And if you have enough of them jammed, then you sort of get an aggregated state that's long-lived. Myxococcus, right, as you saw, you form these plus 1/2 defects, where there is a slowing, but it's accompanied by coming out into the third dimension. So in fact, the cells don't really ever stop moving in that sense. So there are relations to the motility-induced phase separation mechanism, but the real mechanism underlying it is very, very different in that sense.
Great, thank you. Let's see, we've got another question from chat. Buddhapriya Chakrabarti asks, are there chemical signaling that these organisms are sensing?
So yes, on many scales, and we don't think so on some other scales. So this is all induced by starvation. That's a chemical signal, right? I say there's not enough sugar around for me to grow, so I stop growing and I do this stuff. There's also, there appears to be cell-cell contact-based signaling that the cells use to talk to each other. There does not appear in the Myxococcus system to be signaling over long range via diffusible chemical signals. There are other active systems such as dictyostelium or other kinds of epithelial systems, epithelial cell sheets, embryos, for example, in which long range gradients of different chemical concentrations give rise to different sorts of behavior and coordination. And that does not seem to be happening in the Myxococcus system, from what we can tell.
Great, thanks. It looks like we have a question from the panel. So, Eleni?
So I have a little bit of a crazy question. So we've seen in your talk and Christina's talk this beautiful work about defects, and definitely how biology, how life reacts to it, right? There is a defect, things are gonna organize around it. But I didn't see very clear evidence, and I don't know if there is any, that actually biology has evolved to utilize it. I know it's very hard to prove it, but perhaps if there are two separate but closely related species of microorganisms that do a different thing, and maybe one use these as control centers and the other one maybe as more disordered, so there is not the information that there is a defect is not that clear. I don't know. So do you have any crazy response to this crazy question?
I don't think it's a crazy question. So fruiting body formation is absolutely key to the survival of Myxococcus xanthus in the wild. In fact, essentially at all times, almost all the Myxococcus on the planet are living as spores inside fruiting bodies. And from what I can tell, if you disrupt either the motility or the nomadic alignment, you cannot make fruiting bodies. So to me, that is a strong indication that evolution, sort of drove the adaptation of this mechanism, or the development of this mechanism. I don't know. That would be my answer. But I don't know, maybe we don't have other species of mixos. There's lots of species of mixos. They actually almost all form fruiting bodies. It's one of the definitions of being one. And they all seem to do it via motility, but beyond that, I don't think we know too much more about them except the specific one that I talked about, Myxococcus xanthus.
What about other species that make fruiting bodies, but they don't use defects? I think maybe Dicty is one of them, right?
So Dicty, so it's kind of unfortunate that we call these things fruiting bodies. It's just like the name flagellum, right? The bacteria flagellum and the eukaryotic flagellum have the same name, but they're like so different from each other. One's a completely inert polymer with a motor at the base that spins it around. And the other one is this fantastically complicated active complex with polymers inside and motors all up and down the length that have active force generation along the length of the flagellum. Yet we call them the same name because no one knew this when they named it. Fruiting body is kind of the same thing. We know a lot about dictyostelium, fruiting body aggregation, fruiting body formation, slug formation, etc. That all involves very, very different physics at very different scales than Myxococcus. And the fact that they have the same name is, I think, kind of unfortunate. But.
All right, well, thanks again for just a fantastic talk. That brings us to the end of the first session of this workshop. So we're gonna have a short 10-minute break, and then we will be back at 11:35 with our next round of speakers. So looking forward to continuing this discussion real soon. Thanks.
Yeah, the iPad is no good. All you see is your little, it's very panic inducing. But you could hear okay?
Josh, can I ask you something?
Of course, Cristina.
I actually was trying to try something in the chat, but I got stuck. I mean, in the YouTube channel, somehow it's stuck. I don't know, for me at least. It's true that what happens in Mixo is different from MIPS, but in some sense, if you are allowed to escape into the third dimension, crowding will drive escape into the third dimension. And then if you do that, it's true that even though there is crowding, things will still be able to move.
I agree, but they don't slow down at all.
No, I know. We talked about this before. But I
know. So like, I don't know. And the resolution of small aggregates does not have to do with, reorientation of the motility vector or anything like that. So I don't know. And sometimes I kind of agree. If you stare down, if you collapse the third dimension into 2 dimensions, then the effective, diffusion coefficient of a cell in 2D is lower because it goes up into the aggregate and it's kind of stuck in the aggregate. But to me, that doesn't sound so much like MIPS, but I don't know.
Yeah, no, what I was saying is that, in some sense, escaping the third dimension is another consequence of crowding. So,
okay.
They're not such different. Anyway,
that's fine. I'm willing to go with that. That's okay.
Any advice for why am I stuck in the YouTube channel? I can't.
Did you try just refreshing the page or whatever?
Maybe. Okay, I'll try.
Is something wrong with YouTube, you guys? Or is that just a connection issue?
No, it's not my connection.
Mm-hmm.
I misunderstood circular for acircular, but I think I answered the right question anyway. Daniel, I was going to make a little snotty thing about how we do take quantitative data, and that when physicists study biology, it becomes quantitative. Except I realized that my talk was actually very unquantitative because it's only 10 minutes about all this stuff. So I thought making that point was a little bit weak at that moment. But it's true. Like the idea that biology is qualitative and messy is only because the people who measure things didn't do it quantitatively and, you know, didn't do a good job. Like, you know, I've yet to encounter a biological system where you can't make good measurements, right? Like if you wanted to,
right?
You know, and no one's ever convinced me that it's impossible to do so.
Right? I think that's fair, and I think, you know, you should feel free to be, you know, make whatever comments you wanted.
No, I just thought it would
happy to be,
you know, sound a little silly when all I was going to show pretty pictures, right? Like I had like 2 graphs.
Exactly.
I was like, oh, maybe I shouldn't.
But yeah, it was a little bit of an unfair, you know, framing of the question from us in the first place, and kind of did that on purpose. But
I know, I could tell.
Yeah.
Oh, good.
Yeah, in the chat, Margaret Gardell says that she could give you some systems that are hard to measure. So
I disagree. Okay, wait. Hard to measure, yes. But all science, all physics is hard. You really think that like condensed matter measurements are easy? Of course not. They're extremely difficult. Right? But that doesn't mean that you can't do it if you given time and effort and motivation. Right? So it's not easy. You know, like, I don't know. Gravitational waves are not easy to measure. You have to be very motivated to do it, but you can do it, right? Too many windows to pay attention to. I don't think I can do it.
Yep.
And, you know, if I had my real computer, I could have both, but I unfortunately can't. So there's an interesting question in the chat. Maybe we can all chit chat, those of us who are here. It says, when do you expect a living system to fall out of the framework of soft matter? And I must admit that I don't know what the bounds of the framework of soft matter are. I mean, if you tell me that it's about, can I make continuum theories, that I could understand the bounds of it. But there's other, yeah, I don't know. So are there any comments from people who really consider themselves hardcore soft matter people?
Well, maybe in some sense, I think this was alluded to in one of the questions raised in the conference description, in the workshop description, I think, which is, I don't know, I mean, I could try to say something, it's kind of off the wall, but you know, in soft matter, we are used to identify relevant fields based on symmetry and conservation laws and stuff like that. But in biology, what matters is function. You know, things are evolved to perform a specific function. And so we need to start thinking about new ideas, new ways. So somehow, if we want to push continuum description, maybe they should be, the choice of fields should be somehow related to the fact that biology tunes itself to perform specific functions. Now that's kind of meaningless overall, but I'm just trying to. So that's one thing that I think is really missing from soft matter and is there in biology.
Mm-hmm.
You are muted, Josh. Hard to be.
I wasn't actually talking.
YouTube channel, but you're muted on the Zoom. So since we can only keep one.
So Josh, I mean, if a biological system has, you know, regulatory systems and so on to constantly adapt the soft matter properties of your system, I mean, it's still a soft matter system. It's just
exactly.
There is regulation going on. So soft matter puts a lot of constraints on what could be going on, but there's like this extra feature which you wouldn't have in.
But why isn't that soft matter?
Standards and I think it is soft matter physics. It's just that it's a little different from an artificial, you know, like a bunch of colloids or something, because they can.
So sense, that I have no argument.
Adapt, they can regulate.
But you know, it's just not clear to me that the bounds of soft matter have, you know, that there are hard, clear bounds to soft matter, and you know, biological things seem to be inside of the bounds. So like I don't know.
No, but there are certainly things that we don't incorporate. I mean, I can certainly imagine incorporating adaptation and self-regulation and feedback in soft matter models, the way we use them now. Optimizing for function, it's harder, or having function drive your choice of the effective description. I guess that's what I'm trying to say, that I don't know how to do. Again, I see Chase talking in the, what am I doing wrong here?
Should we start?
I see Chase talking in the YouTube channel that I have muted, but I can't hear him on Zoom.
Okay, so welcome back, everyone. We'll go ahead and get started with the second session of, we're streaming, right? Everything?
Like a bunch of...
Okay, so we'll get started with the second session. I want to thank the first session for great talks, very engaging and interesting talks. And I guess just a quick reminder of the next screen. Daniel Weissman will be co-organizing the next session. So just a quick reminder, August 18th and August 25th. The format will be a little different. Talks will be a little bit longer. And stay tuned for more details. You can check out the Emory TMLS website and also follow us on Twitter. And so with that, I want to start the second session. And so if you haven't noticed, we're sort of trying to understand or talk about how biological systems and soft matter can inform each other. And so as we sort of move through the second session, you'll see that the systems are maybe getting larger. And so we'll move to some organismal things towards the end of the session and hopefully have a very nice connection and discussion throughout. And so with that, I want to introduce Michael Brenner from Harvard, and he's going to talk about synthetic living systems. So with that, Michael, please.
Okay, thanks. Can everybody hear me / see my screen? Yeah? Okay, so so thanks for having me, and thanks for having this wonderful workshop. Let's see. So I thought maybe even though this talk is only 10 minutes, I thought it would be better to get to the end before the beginning, and I'm going to do it in a sort of brutally honest way. So this talk, we've been trying to do what I'm about to describe for some time, and basically this talk is really about, the inability of soft matter physics, at least in our hands, to emulate biological function. So it's sort of like the opposite of the title of the symposium. And then I put in help here because there must be a way to do this, and we've been working hard at it, but we would love ideas, etc. So, okay, so that's the end. So now I'll go through the beginning. So the question that we're interested in is to understand what would it take to create synthetic materials with the properties of living ones? And the point is that you all know very well that nature creates soft materials with far more intricate and dynamic properties than we can. And the question is why? And the sort of theory of why is that that's because nature is able to build stuff easily that we can't build. There are synthetic protocols for building proteins, lipids, et cetera, sort of on one hand that is sort of harder, at smaller scale, and with more specificity that we can do. And secondly, because as the theory goes, that these building blocks are programmable so that one can build aggregates out of pieces that are themselves have tuned interactions between each other, and that can be done in such a way that it far exceeds the functionality of just a random set of stuff you would throw in a jar from soft materials. And so, there's a list of things that biological function can do that, it's sort of hard for soft matter. And the question is just what could we do to get there? And the sort of counter argument as to why it's hard is that at least if you believe that these are the reasons that for why for why biological systems are so much more complicated than the ones that we deal with in this community, it's because this programming is not only manufacturing, but the programming is honed and tuned through billions of years of evolution. And how are we going to figure out, right? It's not only what can we build, but like how are we going to figure out how to make the choices to actually build stuff. And so hold up my screen. Okay, so and there's the requisite list of examples of things that biology builds that I will spare you of, that are all soft matter systems. And so our goal, and this we've been sort of working on for a number of years now, is really to try to figure out what are the minimal ingredients for a synthetic material to have these properties, to do it in such a way that it obeys the laws of physics, and that more and even more precisely, what would be wonderful is if one could figure out a way to do it so that it could be actualized in the lab and that the materials could actually be made. So that's the sort of story, and the sort of the thing that sort of catalyzed this is really the tremendous experimental advances that have occurred in the past 15 years, 20 years even, for designing components with specific interactions with each other. So one can build things out of DNA, one can coat DNA on particles, as Chad Merkin has done for a long time. And the picture on the right is from Sharon Glotzer, a review article about like manufacturing of, how manufacturing of particle shapes has come along. And the thought is just that we should be able to figure out how to push all this stuff together to make, soft materials with enhanced functionality, and how can we do this? So we actually have a number of different projects for that have been trying to go after this, and what I'm going to do is just give a high level description of like what we're trying to do right now, and then just to give a couple in the time that I have, small vignettes of examples. So what we basically have been doing is to sort of set up what I think are really great collaborations with experimental groups who build stuff, and then to try to figure out like what are the constraints of what can be built, right? And then to try to figure out, is it possible to like somehow optimize over the set of things that can be built to make something that would actually be interesting. And so colloids are sort of an obvious target for this, although it's not clear what one can do with colloids. So we've been, over the last couple of years working a lot with David Baker's group at the University of Washington, who really has just an incredible ability to design interactions between proteins, even de novo. And they, do remarkable things with them. But the thought was that, well, maybe, if one could design, one could do more. So one of the things I am going to tell you about today is we've been working with Itai Cohen and Paul McEwen at Cornell on what they call magnetic handshake materials, which are sort of physics-based ways of building specificity. And finally, RNA, DNA type systems. So, okay, let me, I'm going to tell you 2 quick vignettes. I have 4 minutes according to my clock. So this is going to be very quick, but just to sort of give you ideas. So I'm going to tell you a little bit about just magnetic handshake materials, which is this thing from Itai and Paul and Chrissy that I will tell you right now, which I think is really amazing as just a system. And then I'm going to tell you a quick theoretical idea that we've been trying to go after, which is about how one might design kinetic landscapes using modern computational tools. So, okay, so magnetic handshake materials. So of course, to build things, we need specificity. Like if you want to build something in equilibrium, which has some complicated structure, then the game is to basically figure out how to make building blocks stick to each other in the way that you want. And if you use like proteins, then you have to design protein interactions. If you just use colloids, you have to sort of like pick up stickers that make this happen. So, so Paul and Itai and Ron and Chrissy had this amazing idea for doing this with a magnetic-based system, which I love because it's entirely physics. The programming can be done, at least in principle, and it's entirely physics-based. And here is their idea. So, so what they do is they take magnetic dipoles. So these are dipoles which are like, up or down. And so this is a pattern of dipoles. And then what they do is they embed them in a substrate to make panels which stick to each other. And the beauty of this is that one can, of course, do this macroscopically. And, they have, so Itai and Paul have experiments doing this macroscopically, but one could also do this lithographically and thus, build magnetic dipole patterns on blocks really at a much finer scale on small systems. And this is just like an amazing template upon which to build interactions because, you can put the up and down dipoles wherever you want, basically, and try to get things to stick to each other. And, they're magnets, so you can put on fields. There's just like a ton of stuff that you can do, experimentally. And so we've been trying to think with them about, how one would design these to make useful things. And so this is, Chrissy and Itai and Paul and the crew, have sort of lists of things that they've sort of imagined. And, there are sort of experimental realizations right now in shakers with particles, but just to sort of give one idea. So the question is how, and this is, I'm going to accelerate through this because I'm now talking slower than I should. So the question is how does one choose? So how does one choose the set of dipoles on a face? And so, you can, maybe I'm going to skip this slide. So I'm going to skip this slide too. Sorry, this is math gone. So here, the question is, so, you can put the dipoles wherever you want on these faces. You can put any number of dipoles and you can put them with different strengths, and you can put them in different locations. And how do you choose sets of dipoles so that you actually have interactions which stick to each other but don't stick to the other guys? And this is a sort of question that we've been working on. And what's nice is that it's entirely physics-based. I mean, one can sort of write down formulas for the interactions, but the fields that are produced from these different interactions, and then just try to literally do this by optimization. And I don't have time to go into this. These are slides that aren't going to exist. I'm sorry, this talk was less well organized than I had anticipated. But the point is to try to come up with actual designs. And Chrissy has been doing this in her computer and theoretically to try to come up with actual designs of dipoles. So this is an example of 3 dipole patterns. These are 10 3-dipole patterns, which are more or less orthogonal. So this is the binding energy of like this dipole pattern to this dipole pattern. And the idea is to somehow design them by like just building them on the faces of the thing, and then to build assembling systems in the lab, which basically emulate them. And so this is sort of one direction. And again, what I guess I find nice about this is that it appears to be a very rich system for designing interactions that can be then used to build interesting things. And so and these were going to be simulations that show that designed things can do better than random and blah. Sorry, I'm going to skip these slides. This was a badly organized talk. These 10-minute talks are tough. You guys really, I should have practiced. This is a huge mistake. Anyway, okay. So just one more idea. This is 10 minutes, so I'm going to, I'll take 2 minutes after the questions. So another thing we've been thinking about, this is just a broad thing, is designing kinetic landscapes. And And I mean, the point here, of course, is that, you know, designing equilibrium systems is sort of boring. I mean, like, you can make, especially if you do it the way that I just described, which is that you just build locks and keys so that the locks stick to the keys, they don't stick to the other ones. You do the best you can given the constraints that you have, and then you hope for the best and you try to optimize. I mean, that's fine, but it's a bit on the boring side. So the question is, can you design, you know, what about kinetics? And just as an example of this, so this is a paper that Carl wrote a couple of years ago about designing gels. So this is a particle, this blue particle is designed to basically diffuse through this gel. And the way it does it is that when the blue particles interactions with the green and the white particles is designed to be exactly so that when they bind, that by diffusion alone, the blue particle can break the bond. But if you choose another particle, which doesn't have exactly the right binding energies with these other things, that it basically gets stuck. And so this is a sort of kinetic state cross-link state diagram for the blue particle with the green and the white particles. And here, if it will play, is a movie in which you see that the blue particles, which were designed to interact specifically with the green and the white particles, you know, are able to get through the gel, whereas the red particles are stuck because they can't break the crosslinks. And so this is an example, a simple example of a soft matter system with designed energies in this case that leads to a property that is really much more spectacular than could be achieved, you know, just by random guessing. But this is only with 5 parameters. I mean, that is the parameter space here is really very small. And, you know, one could imagine, I mean, if one looks at biological analogs of this, for example, like, I mean, we were inspired originally by the nuclear pore complex, like we don't even know how large the parameter space is, but presumably it's much larger than this. And how would you go about doing this? And so, so I guess so sort of motivated by this, we started looking into sort of computational ways of searching for kinetic pathways. And I'm now really out of time, so I'll just say one idea. And so what we've basically been doing is the following thing: so there's machine learning, which is everyone's up in arms about in these days, but there's also the tools of machine learning. And one of the critical tools of machine learning, which I think is very interesting for this purpose, is automatic differentiation, which basically allows one to efficiently compute the derivatives of functions which are expensive to evaluate by not having to do a function evaluation every time you want to take a derivative. So if I give you a function of n variables and I tell you to please differentiate it with respect to each of the n variables, naively, you have to evaluate the function 10 times or n times in order to do this. But there's a sort of classical technique from control theory, which is called the adjoint method, which allows one to compute all the derivative of this function with respect to all of these variables only using 2 function evaluations instead of n. So this is the reason that, so this idea, the adjoint method, is sort of hard-coded into all modern machine learning languages. In fact, it is the primary design criterion, is easy of use for this. This is TensorFlow, PyTorch, JAX, whatever, and this is called backprop in that context. But the point that I want to make, though, is that this technology, that is the ability to sort of compute derivatives of very complicated functions, is extremely useful for physics. And in particular, if one is interested in doing simulations, for example, to try to optimize parameters to get things, you know, the function evaluation might itself be a molecular dynamics simulation, which is very expensive. And so you would never, right, you know, sort of searching a large, searching a large parameter space is very complicated. And so what we've been doing is trying to use the tools of machine learning to try to do these calculations in various examples. And I don't have time to go through this, so I'm done. And this is sort of inspired and is in collaboration with Sam and Dosh, who both work at Google Brain, who were soft condensed matter physicists, in fact, who wrote this wonderful, just to pitch for them, package, which is called JAX-MD. It's a molecular dynamics package, which is fully differentiable. And so one can play these games. And we've been working with them, trying to basically design systems and kinetic landscapes and whatever. Anyway, that's the end. I'm sorry, I have too many slides. That was a badly organized talk. I'm going to end with this. And thank you all for your attention.
Great, thank you very much for an interesting talk. So the question I have for you is, what kinds of biological hypotheses do you think that you could test with these systems that you're able to design, these experimental systems?
I mean, so that's a good question. So you could certainly use it to test biological hypotheses. Honestly, at the moment, we're being much cruder than that. I mean, we would love, for example, I mean, like if you take the gel as an example, like, I mean, there have been debates, about the biology of, of how nuclear pores work and how basically the selective gels work. And this has been, debated by biologists and physicists for a long time. And the sort of creating a system which is built out of simpler components, which has that functionality, doesn't necessarily shed any light at all on that issue. Like that is the gel, Carl's gel that I showed you might actually have something to do with biology, but it may not. And in fact, that's not our point. Our point is to just try to find out, is biological complexity a result of, the large number of parameters and the fact that there are small parts of parameter spaces with very useful and interesting functions. And if so, can we figure out a way to create them with materials? And I mean, one would hope that says something about biology, but that's not in any sense our goal. We just want to figure out if we can build interesting stuff. That's sort of evasive, but it's the truth.
Sounds great. I think Daniel has a
Sure. I mean, beautiful talk. And just following up on that a little bit, so a lot of what you talked about was this, just beautiful work on designing specific interactions in systems that, the microscopic unit was at different kind of characteristic length scales. Do you think when we think about designing synthetic living matter, do you think there's an important distinction between what length scale you're starting with? Or in your mind, is it just a question of the complexity of, the ultimate phase space you're able to access?
That's a good question. I mean, I guess, well, it should probably be small enough to be Brownian, right? So that, Brownian motion is important. So that's sort of one comment. I mean, beyond that, there are, I'm not sure what you're referring to. There are, in principle, right, a Langevin equation is a Langevin equation. That's sort of one answer to the question. On the other hand, if you're interested in building things with hierarchical structure, then that's another thing you could imagine doing. But as soon as you say hierarchical structure, then that's immediately, making statements about kinetic pathways, because if you're going to build something hierarchically so that there are different scales, right, that are interacting with each other, if the whole thing hasn't equilibrated, then you need to somehow program that in. So you need to program in pathways and in some way. And I think that's when there's a huge phase space of possibilities. There's lots of stuff one can build, but it gets hard very quickly. And I think that's the reason for this statement on this slide. I really do think that there's a lot of potential here. Even for doing very, very, very simple things, which is partly why I like this. But on the other hand, what we can do with the components that we have, is a difficult. It's difficult to figure it out.
great. Okay, so we have a question from John Johnson. Are these components programmed, or has there been evolutionary selection for the emergent behavior of the components' interactions? Is this a valid and necessary distinction?
I'm not sure I understand the distinction. What does it mean, program? Program means you've chosen if you choose the interactions, if you choose how things interact, I can call that programming, or I can call it the result of evolution. I'm not sure I understand the distinction. That might be my lack of understanding, though. Probably it is. Sorry.
Maybe John can elaborate in the chat and can follow up with that later. And I guess with that, I want to thank you again for a great talk, and we will move on to the next speaker, who is Eleni Katifori from the University of Pennsylvania, and she will be talking to us about information in fluid flows.
So I hope you can see me and hear me okay, if not, holler. So before I begin, I would like to thank you guys for making this happen, for organizing this for the community, and for inviting me, of course, because this is an island of sanity in this these crazy times. So before I begin the talk, I would also like to show you a picture of my group. This is the last pre-COVID picture, and some people couldn't make it, but also there's been some changes. So you see there is a lot of photoshopping I had to do.
I think you still have to share your screen.
I am. It says here that I'm screen sharing
Got it now.
my Zoom. Okay, so there was some delay. Okay,
Yeah,
good.
sorry, it was my fault. I had to turn off mine. I apologize.
Okay, good. So on the slide here, you see my group. And as I said, it's been a bit of photoshopping that needed to happen, but I would like to thank them also very much because we discuss a lot and they're all very helpful in basically guiding the direction of the group. So our general interest, among other things, let's say our primary interest has to do with biological flows. And in particular, when these flows happen through networks, right? Plants or animals, because it's a very beautifully complex system. And as I will try to convince you, it's very, very important for many aspects of our survival. And also a particular aspect that we're interested in is the connectivity and the topology. And we have many questions that we've been dealing with recently. For example, how do networks develop and how do networks self-organize to hierarchical structures? I know very different system than what Michael was saying, but it turns out that biology knows how to do it very well. Now, there are many, many things that we're curious about, but more and more recently, we care about dynamics. We care about how the network dynamically responds to changes, and that brings us to what we're going to talk about, which is information transmission through fluids, basically. So the first thing that I should say about this is the obvious, that water is necessary for all life, and life evolved in an aqueous environment, right? And physiology actually is all about regulating water, solutes, the temperature, so that the cell has conditions that are compatible with life. So water is super important and inescapable. Also, information transmission is necessary for multicellular life. And here, when I say multicellular life, I mean it kind of broadly. This could also be like ecosystems of unicellular organisms that basically function together. And they need to integrate signals from the whole body and make decisions that optimize the global fitness, right? So when we hear information transmission in biology, we mostly think about action potential and electrical signals and neurons and all that. However, this is not the whole story at all. There is a lot of information that is transmitted via the fluid medium. And there are two ways. Again, the obvious way, and that is by propagation of signaling molecules and solutes with the flow. This is obviously hormones, right? So you maybe something happens, you get scared, you have adrenaline being released in your system, and things change, right? This is not a very efficient information transmission pathway because of diffusion, because of dispersion. And that is, again, there are some physical limitations that maybe soft matter will help us put in some physical limits on whether you should use it or not. Now, there's some very interesting cases like Physarum polycephalum. We're not going to talk a lot about it, but actually this was a paper co-authored by one of the people that spoke today, where basically there's a feedback loop of the signaling molecule to the propagating contraction front, and that is so that you maintain the transmission of information longer. But it's again, it's you should still classify it more or less as a chemical transmission of information. Now there is the not so obvious way, which is the propagation of mechanical energy via fluid structure interactions. And I'm going to talk about two things here. One is not work of my own, which is protista in lakes and ponds that communicate via predator evasion. And what I will talk about, though, is a very small and very brief snippet about back of the envelope calculation that has to do with propagation of arterial pulses in the human circulation, right, our body, and which is necessary for pressure homeostasis, basically for us to, regulate our pressure and remain alive when abrupt changes happen to the pressure. Now, what you see on this slide is the life adventure of Spirostomum ambiguum, which is one of the largest protista, which usually swims at low Reynolds numbers, but can rapidly contract. And while doing so, it releases toxins. And this rapid contraction generates long-range flows. Now, a cell detects predators through the surrounding flow, contracts, and in turn generates flow that communicates that information that there was a predator to neighboring cells, which also triggers them to contract. And this way, with this wave, the whole population collectively releases enough toxin to make the predator go away. So this key thing here is hydrodynamic coupling. And this work was actually done by Arnold Matthijssen, who used to be at Stanford, and now he's actually coming to Penn winter 2021 as a junior faculty. So we're very, very happy. So here we have transmission of information through a continuous medium like water. But now I want to talk to you about something that pertains to our body, and that is pressure regulation, right? What happens is that what you see on this picture here is there is a person and they put inflation pumps, cuffs on the thighs, and they kept them inflated for a very long time, and then they suddenly release them. And what you see here is that there's a sudden drop of blood pressure because the hemodynamic profile that the body has adapted has changed drastically and very quickly. And you see a sudden drop, but not instantaneous drop, right? There is a time scale here. And then somehow the body figures it out that now these legs that weren't doing anything are now back in business. And things adapt. There is vasodilation and vasoconstriction, and the pressure goes back to regular. So there is this, in a way, mechanism to adapt the diameter of your vessels, and there is this drop here that we're going to be most interested in and we're going to be discussing in the next slides. Now, the important thing, the key to this question is that our vessels are compliant. This has been known for a very long time. There's a lot of done here, and if you want to Google it, it's the Windkessel model. And every time the heart contracts, there is a pressure and volume wave propagated through the system. And one could take the Navier-Stokes equations, and we did. You can integrate them out to make a reduced model, which actually turns out it looks very much like a transmission line, right? So if you like E&M, this is your game, right? And the flow is viscous. The fact that the flow is viscous is represented with a resistor, the inertia with an inductor, and the elasticity with a capacitor. And I can take these quantities here and I can define from them some dimension full quantity, some characteristic length scale, a characteristic time scale, and a characteristic admittance that depend on the physical parameters. And from the length and the time, I say, okay, I will get a characteristic velocity, right? I can get a characteristic velocity by dividing lambda over tau. And the question is this actually how the velocity of the information that a pulse is coming or that the heart rate changes? Is this the actual velocity? Well, now let's, to answer that question, let's do some calculations, right? So there is, let's assume there is an abrupt change of flow that happens at the pump. You are not doing anything, you start running, right? Then there will be a current wave that will propagate through the system with a particular speed. And actually the speed that this front propagates determines the mechanical information transmission. And there are two actually separate time scales if you look at it carefully. One is what I can call T0, which is the earliest time that the current at a particular distance z along this 1D vessel begins changes, and that is noise limited, right? And then it is something that I call T50, the response time, which is the time it takes for the current to reach the 50% of its final value. Now, this T0 time, which actually scales than 1 over v, the velocity I told you earlier, is useless to the body because that's very noise limited, right? Whereas the 50% is the actual time scale for the information transmission. And so there is some actually very elegant calculations involved by Sean Fancer. And in the end of the day, in the limit of long vessel, what we have is a very different time scale than the one that we predicted earlier. Actually, a time scale that tells us it's not the inertia of the fluid that determines how fast this pulse, how fast the information is propagating in the system, but it is actually the resistance of the vessel. right? And the capacitance, which is basically the how elastic, how compliant the vessel is, also plays a role. So of course, the surprising thing is that you can do this for networks, and I won't say a lot, but it actually works. This can be generalized for very complex networks. I'm showing you a simple one here because this is what we did the movie for, but it works for many different kinds of complex networks, this estimation of the velocity. And there is another fact that I will mention very, very briefly because I don't want to take a lot of your time, is that actually, if you want to design a system, if let's say you are biology and you want to evolve a system with very, very fast information transmission, so that you are able to figure out that maybe you got injured and you're losing blood somewhere, or your head is upside down, or I don't know what, then if you want to increase that information transition, then for the response time, if you want to decrease the response time, then you have to pay in dissipation, right? So there is a cost-benefit analysis here. And surprisingly, again, the scaling, there is a very robust scaling relationship between the dissipation and this response time, which is a square root relationship, which again carries over many, many different topologies of network. It is very, very robust. Now, the last thing I want to tell you is this theme of so far you've seen that if you try to transmit information in via solutes or via this mechanical information transmission, you will have the front of the pulse, the sharpness of the pulse, basically, of the information pulse getting attenuated. The pulse broadens through diffusion, or in our case, through, again, these processes that I was describing. Now, we know from neurons, right, that excitable systems can propagate a soliton that maintains the shape of the pulse. This is a Hodgkin-Huxley action potential, and all excitable systems can do it. So fluidic system seems doomed from the get-go, and it seems that they won't be able to do that, but actually Miguel Ruiz Garcia, which you see here, he did this very nice work where he's shown that you can actually construct a Hodgkin-Huxley-like fluidic system that behaves like an excitable system and propagates it propagates the information basically like a soliton without dispersing it. So you can see more of that in these two papers that hopefully are going to be out soon. So with that, and not going too much over time, instead of me asking you something, I would like to ask you something, right? Given the theme of what soft matter can do for biology, and perhaps vice versa. So I showed you this network systems, this network system information transmission in network systems, but can we, off the top of our hat, basically figure out with back of the envelope calculations what the physical constraints for whole organisms integration, much like the time scale that I showed you earlier, and what are the costs and benefits of each mode of communication? Where should I do solute and when should I do mechanical? And perhaps more importantly, when are neurons necessary? And when can an organism get by without them, right? Do you always need a nervous system? For what do you need a nervous system? Can soft matter say anything about this? I guess so with that, I think I have to stop. Otherwise, I will be kicked off by the organizers.
Great, thank you so much for a wonderful talk. Just out of curiosity, does the panel have anything that they want to comment on regarding these questions? And if not, I would be interested to hear some of your thoughts on these questions.
So I would, that's a good question. And in fact, I would borrow something from Michael, and I would put a help with an exclamation point, because our expertise is in networks and fluid networks and how to bring soft matter into fluid networks via fluid structure interactions. But there is a whole lot we don't know anything about, which is neuroscience in a way. So I guess I can calculate again from an electrical like system the action potential propagation speed. But what are the costs to putting ion channels? What are the what are the costs of making the, I don't know, the conductance of the fluid in this next to the, in the action faster or slower. I don't know, right? So this is, I can only answer this in my narrow universe.
Okay, great. Do you have any comments on the kinds of energy needs that would need to be put into the system to sustain unattenuated information transfer?
That is actually a very good question. So there is, it depends on how you actuate it, right? It depends on how you actuate the what you need, right? You can do it actively or you can do it passively. So passively, there would be some, perhaps there could be an instability that would tell you if you push flow, flow through a particular pipe, if you have the pressure drop through the pipe increasing above a certain threshold, the diameter will decrease. There's various instabilities that could do that, or you could do it actively. And in fact, it looks like in our body, the contraction happens via the smooth muscle, right? So we have all our vessels are aligned with smooth muscle, which actually, all our arteries and arterioles and so on and so forth, which actually react to, they measure the pressure basically, and they actively react and respond. So I could imagine a mechanism that happens with some sort of chemistry which is beyond our purview here, that reads a particular pressure drop signal and acts. And the energy cost there would be, I guess, the metabolic cost of the cells to do what they're supposed to be doing.
Great. And then a follow-up question from the chat. Ilya wants to know, what do you mean by information? When you say information is being transferred, what do you mean?
Yes. So what I mean is the following. So let's make a very, this is a little bit naive and extreme, but it will make the point. Let's say for some reason, my heart starts pumping at time 0, my heart starts pumping faster. The vessel at my toe will not know that. It will only know that after some time has passed. I call it information because it is something, so the whole body needs to be integrated one way or the other, right? And so that the whole body will have to find a new steady state. The whole body will have to react to this new pressure profile that is being pushed in by the heart. So the fact that you have this mechanical wave essentially traveling from the heart to the toe, that you could argue that this is information at the toe, which says, okay, now the pressure is not what you thought it was, the pressure is different. And then based on that, the vessel can take an action, maybe decrease the diameter, increase the diameter dependently. Now this competes, so there is, I call it information, I don't have to call it information, but there is actually a competing, this is a very good question because I can mention something now that is very important. The way this whole body regulation happens is not entirely clear right now. So there is the parasympathetic nervous system, which actually we used to think that that's one of the primary ways that you have this whole body integration. So the signal that something changes somewhere used to be, can also, the vasodilation or vasoconstriction can also be regulated with neurons. So when the neurons transfer the signal via an action potential, I call that information. So I also call it, call the fact that the pulse is traveling through and the information is actually directly transmitted via the vessel. I also call that information, right? I don't know if that answered the question, but I'll check the chat later.
Okay, great. Thank you so much for so we'll move on to our next speaker, Symone Alexander. is a postdoc at Georgia Tech currently, who will be starting her own lab in chemical engineering at Auburn in January. And she's going to tell us about multifunctional materials designed and inspired by nature. So Symone.
Right, thank you, Jennifer. And thank you to Daniel as well for inviting me to participate in this really exciting workshop. I've been thinking about research and design at the interface of soft matter and biological function for a while now, so it's really exciting to participate here. So right now, my research interests really encompass this idea of reverse engineering biology for materials design. So I'm very much an experimentalist, but a lot of what I do is informed by applying soft matter theory and kind of fundamental engineering concepts to biological systems to then design macroscopic composite materials. So this has incorporated everything from looking at how polymer chain dynamics influences self-assembly of small molecules. You can see kind of similar things when you think about our skin and thinking about cholesterol assembling next to collagen. And then also in designing bio-inspired composite materials, so using seed pods and feathers as inspiration for these responsive composites that actuate in response to changes in the environment. And then more recently for my postdoc, I've been looking into some soft matter biophysics. So spiders are some of my favorite engineers, and they use their silk for some pretty cool processes. And I'll tell you a little bit about this cool spider later on in the talk and why its silk may be even more important to the discussion. So what exactly is this idea of bio-inspired materials design? Nature is really great at designing materials with multifunctionality, which is kind of hard for us to do as scientists and engineers. So you can think about things like our own extracellular matrix that can respond to temperature, damage, things like that, or something like the Hercules beetle, who can change its shell color in response to humidity in the environment, or something as humble as a seed pod that, again, is responding to moisture in the air in order to improve its chance for survival. And so a lot of what I like to do in my research is connecting these biological functions to applications of materials. So creating release agents, sensors and switches, things like that, or actuators. And so typically nature is using structural hierarchy of soft matter to achieve these things. And so how do we then connect this biological function to materials design? If we look at these biological systems and the fundamental concepts that they're using, you see a lot of recurring themes. So you'll see the structural hierarchy, elastic energy storage and release is super important for motion. And then also the use of fluid dynamics. So using hydration as a way to control material properties that then influence that elastic energy storage and release. So they use these things to achieve that responsiveness and multifunctionality. And as scientists and engineers, we can also begin to use these in the soft matter theory that explains the connection between these different variables to both understand these biological processes and design new materials. So this workshop is really exciting to me because I feel like that piece between biological function and materials design is the theory and modeling. And a lot of that is based in soft matter physics. So I'm going to give kind of two examples. It includes some work that I've done with both my PhD advisor and my postdoc advisor, and then the people whose papers we went to all the time. So on the left is Professor Corley, my PhD advisor, now at the University of Delaware. And her group really focuses on mechanical enhancement of multifunctional materials. A lot of these are bio-inspired. And then also Professor Mahadavan is someone whose papers we went to all the time. He has a lot of great modeling and theoretical descriptions of biological systems like pine cones that we used in our own research to then design materials. So starting from the biological function, we've probably all seen a pine cone. They open really slow, so we probably really haven't seen their opening process. So here's a nice little a graphic for you to see that. But on the bottom is an image from a paper by Rieser and Mahadavan, where they took a pine cone and they really described the processes or the biological function of it doing this. And so really pine cones use this motion called hygromorphism to optimize seed dispersal. And so when it's humid in the environment, they are wet, and then they close when it's dry, or sorry, that's reversed, that should be reversed. So they should be open when dry. And that helps with seed dispersal, so helping them getting further away from their parent tree. And so what they did with Mahadavan's group is they were able to break this down into some really soft matter theory type concepts and modeling. So a pine cone in each one of these nodes has an active and passive layer. So in the active layer, it's going to swell in response to moisture and soften, so the modulus will change. Whereas there's a passive layer that's attached to that is really going to maintain its modulus, so it just acts as a reinforcing layer. What's cool about what they did is in this equation here, they've connected the responsive curvatures, so how these pine cone change or open up to the expansion coefficients of each of these layers, so how much they swell in response to humidity, the change in moisture content in the air. And then this term F of Mn is really just a relationship between the layer thicknesses, so the differences between the passive and active layer, and then their elastic moduli. So if we look at this in the model that they developed, from a materials design perspective, we can pull out some key design parameters, and that's differential swelling between two layers, a change in modulus or mechanics of the material in response to that wetting, and then also some structural anisotropy. So in this image here, these pine cones use aligned nanofibers to actually control how much it opens and closes. So just directing that motion a little bit more. And so then what we did in the Corley group is we used these same concepts to create these biomimetic bilayers, where we created some aligned fibers that were interfaced with a reinforcing layer. If you change the alignment, you can then change how these composites bend and form in response to moisture. So it was really exciting because you go from a pine cone that you really don't see open and close to these soft actuators that are also mechanically reinforced, so they're able to do work as well as have this responsive nature. So then the next and last example I'm going to talk about are these really cool slingshot spiders that I've been studying in Professor Saad group at Georgia Tech. And then someone whose papers I've gone to a lot is actually Professor Todd Blackledge. He does a lot of the modeling and theory of energy dissipation in spider webs. So really cool research that we're using, and we're actually collaborating with him now on a modeling paper, which is exciting. So to get back into the biological function, in this case, this is the work that we're doing. So this really cool tiny arachnid, the slingshot spider, instead of hanging out and waiting for prey to hit its web, it takes that beautiful orb web that you typically see and attaches a tension line. And it uses this tension line to extend its web into a 3D cone. And it releases that tension line like a slingshot and flings itself and its web backwards to catch prey. We took some high-speed cameras out to the Amazon rainforest to quantify this and found that it accelerates 2 orders of magnitude faster than a cheetah. And so our biological function here is predation, but our mechanism, which is exciting, is this ultra-fast motion through web extension and release. And so if you think about how these spiders are doing this, is with this viscoelastic silk. And so we can change this, this really cool spider web into a physics problem, right? In a mass between two springs. And we can start to model and understand, one, how it uses the silk for elastic energy storage to get this ultra fast motion, but also how it uses its web structure to rapidly dissipate that energy so that it can actually go and catch its prey and doesn't get flung from its web. So our design parameters that we found thus far is that this silk mechanics are very important. So spider silk is already a really exciting polymeric material, but because this is a new use of spider silk, there may be some new design parameters there. The web structure is also very important. So thinking about how that plays a role in energy dissipation. And then finally, the web displacement. So the spider's behavior, how much does it extend the web and how does that influence how fast it can move? And then as for materials design, you have to stay tuned. We're still developing the model and pulling out the key parameters, but we think it's going to be exciting to build not only materials, but structures that mimic these slingshot spider webs. And so finally, I just want to close with saying that the opportunities for the application of soft matter theory to biological systems are endless. I'm a little bit biased as a soft matter person who applies physics and things to biological systems, but there are some major challenges that we've seen in our research, and part of that is preserving the sample integrity. A lot of these biological materials on the macro scale are designed to be in the environment. But we're moving them to our testing systems. They're not meant to be in a tensile test or in an SEM, so we lose some of that sample integrity. And again, the characterization of those material properties. And then finally, the complexity and interactions between variables. When you're talking about living systems, especially something like a spider, there's a lot of decisions that it's making that we don't understand. So I think some of the opportunities for innovation here are developing some dynamic and portable technology. One of the ways we were able to study the slingshot spiders is through the invention of these field portable high-speed cameras that we could take to the rainforest. And another thing is these messy materials. A lot of times these living systems build things that are good enough to get the job done. And I think in the pursuit of perfection, we've lost some of the nuances in design that can help account for some of the downfalls that we have. And then finally, I think that simulation and machine learning are going to be super important to kind of tease out some of this complexity and interactions between variables and let us know what's it's really important to pursue in our research. And so with that, I will end and say, Jennifer mentioned this, but I will be headed to Auburn University as Chemical Engineering Department to start my own research group. And if you're interested in these bio-inspired polymeric systems, there's some cool resources here that I recommend to get started.
Great, so much for a very fascinating talk. I think Daniel Sussman from the panel had a question.
Oh, I did, and now I see that it was just asked in the chat by Tom Bochenek. Do you have any notion or results from your model regarding what the function the spider web itself was optimized for?
Yes. So we are still working on it, but some of the primary things that we notice are important are, one, the number of radial lines in the web. That's important both for the elastic energy storage, because if you can imagine, if you have a bunch of springs, the more you have and the more you pull, the more energy you'll be able to convert to that kinetic energy. The other thing is that tension line is not only important just as a drawing line for the spider to extend its web, but also for energy dissipation. So it gives you that opposing force that helps the spider slow down. The other thing that we've noticed that we're still working on, because it's a little bit more nuanced because of the structure of the web, is what the material properties of the silk itself have to be. Can you just use any spider silk, or is the spider actually designing for its web structure? Are there specific proteins or amino acid relationships that the spider is using? So we're still working on that one now.
Great, thank you so much. Do you have any insight into when it is more efficacious to explore material properties versus the overall structural design?
Yeah, that is a tough question, because I feel like they're so related. Because a lot of times the structural properties are important to what you're doing with the material. So they almost go hand in hand. Most of these materials and their responsiveness use structural hierarchy to achieve that. So it's kind of difficult to separate the two, at least for me.
That makes a lot of sense. Do you have any sense out of how different spider silk might be from species to species, or is this something that is, do these spiders make something different that is more beneficial for this motion, or?
Yeah. So spider silk is extremely diverse. A lot of the research has focused on orb weavers because it's been shown to be super tough. A lot of it is tougher than Kevlar, used in bulletproof vests. But spiders, even a single spider creates different silk for different purposes. The radial lines that are like the spokes in the web are super elastic, versus the capture threads have these little beads of sticky droplets on them. It uses a different type of silk to jump from plant to plant. So these spiders are changing their silk based on their needs. So, and both for their function and then across species, they have different predation methods. So they also change their silk for that also. So there's a wide range of silks out there, but surprisingly, a lot of it is focused just on orb weavers. So I think there's a lot we have left to discover about spider silk.
Very cool. I think Josh from the panel has a question.
Yeah, hey, Symone, that was amazing stuff. In thinking about bio-inspired design, a lot of these materials are built by like nanometer scale amazing protein machines. you know, that are very different than the kinds of machines we usually use to make things at industrial scale. So is it like, let's say you even understood the structures, do you then look at the machines that build them, or do we have to think about different sort of industrial ways to produce materials that have these kinds of properties?
Yeah, that's a great question. I think in order to translate it to application, we have to start thinking about how we can build them themselves. And that means engineering design. A lot of people, especially with spider silk, have leaned on electrospinning, or even more recently, microfluidic spinning, to try to get the same type of fibers, because part of it is not just, you know, the molecular assembly, it's how it's processed and what that does, like what shearing does to that alignment and assembly. So it's, yeah, it's, I don't think we'll ever be as good as the biological machines, but I think we still have a long way to go, and we can probably do better than we are now to design engineering processes.
Great. Okay, one more question from the chat. In the first example, you showed an inert system, but in the second, there's an actual moving animal, the spider. So although the response is fast, do you think that the spider plays an essential role?
Yeah, most definitely, most definitely. So the part that I didn't show about this is that, especially for ultra fast motion in these systems, they're known as, it's like power amplified systems. It is a series of ultra fast movements. So the spider itself, if you take a high speed camera and you look at its leg motions for when it releases the web, it's moving its leg on the order of microns in less than a millisecond, right? So the spider itself, it's the biomechanics of the spider is just as important to getting this ultra fast motion as the material that it's using as a tool to, you know, fly backwards through the air. So yeah, the spider itself definitely plays a role in this, which I think is what makes these bio-inspired systems and studying them when you have an animal involved even more interesting, because you have to think about their decision-making, their behavior in the design as well.
It's very interesting. Are there any other questions from the panel? Then I guess we will go ahead and move on to our final speaker. Orit Peleg is at the University of Colorado Boulder in computer science, and she is going to talk to us about dense insect aggregations.
All right. Hi, everybody.
It looks like Orit may be having some technical difficulties, but hopefully she'll be back momentarily.
Can you hear me now?
Yes, good.
Yeah, it's great.
Okay. I'm sorry. I just got kicked out of the building for a fire alarm, so the internet connection is going to be super unstable. So I'm going to try and just let me know. Hopefully this works out. So just to confirm that you can hear me and see my screen, can somebody please nod? Awesome, cool. So hi everybody, sorry about that. I'm really excited to participate in this workshop, and thank you to the organizers for inviting me. It's been really exciting so far. As Jennifer probably told you as an introduction, I'm going to talk about a larger length scale of soft matter and materials. So I'm going to talk about a collective behavior. So when multiple organisms basically aggregate together. And I want to make the distinction clear that I'm not going to talk about this more sparse aggregation, like the swarm of midges that I'm hoping you can see here on the left side. I'm going to talk about more dense materials like this honeybee swarm, where the individuals are actually creating these materials while creating elastical or mechanical bonds between individuals. So there's a lot of interesting ideas about sparse interactions, and I think actually Eleni alluded to it a little bit in terms of hydrodynamic interactions when it happens in water, but I'm going to focus on the more dense aggregations. And there's actually a beautiful breadth of functions for these aggregations. Just to name a few, there are bridges that army ants create that I'm hoping you can see here on the left side. The creators to traverse rough terrains. There are ant rafts, so it's rafts that ants create in order to survive floods. Just to name a few that work on these behaviors. We have Symone Garnier, Ian Cousin, David Hugh, Dan Goldman. And then there's also larger scales aggregations of insects. I'm going to talk about these reproductive swarms in a few slides. Their main function is actually for thermoregulation. But then it goes really all the way to bivouacs of ants that actually create them in order to create a nest structure. So this is
so
shown
Orit,
here.
Orit,
Yes.
we don't see anything but the title slide. I don't know if your slides are not advancing in the video or.
Okay. Let me stop sharing and try to share this again. Maybe if I share my entire desktop, that would be better. So can you see some bees and some midges? Okay, so that was just referring to the dense aspect of the aggregations, and now I hope you can see the structures that I just mentioned, the bridges and the rafts and the reproductive swarms and bivouacs. And I was just talking about that last figure here on the bottom right, that Kronauer took actually got some really nice awards for it. So this is an entire nest of ants, basically. So keeping all of these functions in mind, I'm hoping to address two of the questions that the organizers posed. One is, what can soft matter physics teach us about these kind of aggregations? So to answer that, I'm going to show a few examples of these kind of aggregations in a more steady state under dynamical perturbations, and also in terms of their aggregation and growth. I want to point out that there are definitely challenges for studying these new kind of systems. First of all, these are non-model organisms. We don't know as much about bees and about ants as we do about Drosophila, for example, or cell lines. We also need to develop new behavioral assays. And so currently, everything I'm going to describe is going to be really phenomenological. But I think there's a real opportunity there to, first of all, learn something deeper about animal behavior, because now instead of, we can think about a single organism as a building block for these collections, and basically treat it as a black box and think, what are the inputs, what are the signals that this individuals is receiving from the environment, how is it processing them, and to execute some behavior that eventually helps the entire superorganism. And then I hope I have time, I'll also try to pose some, I would say, general call for help, but some areas where I think there is a opportunity for developing new theories that could help us understand the systems better. So I'm going to start with an example of a steady state analysis of the material. These are fire ant aggregations. This is work done by David Hugh, who is really one of the pioneers who started working on insect aggregations. The first thing that they notice is that these materials really display both liquid-like, as you see here on the left side, there's a penny just kind of sliding through this material, but also solid-like and elasticity properties of these materials. They actually did something that people refer to as insect rheology. They put a bunch of these ants inside a rheometer and performed some measurements, and lo and behold, indeed, these materials display viscoelastic properties, which is useful because then theoreticians, for example, people from Frank Werner's lab, can take that information and link it to a statistical mechanics representation of ants and the crosslinks that they make, how often they make them, how often they form the bonds, how often they break them, in order to basically feed these models and learn something about the microscopic behavior of the ants. But what about materials, aggregations that actually experience more dynamic kind of forces that completely change the mechanical landscape that they would experience? So an example of that would be a honeybee swarm that I hope you can see the movie. This is basically a swarm on a tree branch. It's a feral swarm and it's going to be shaken by the wind. So it's going to experience this constant mechanical shaking. And to understand this better, we created one of these or several of these swarms in the lab under more controlled conditions. And we could apply these mechanical perturbations to see the response of the swarm and to try to understand how, what kind of information each individual bee is using in order to stabilize the structure. So the response looked like this. This is a sped up version of the response. We see the front view and the bottom view. The swarm starts as a cone-like structure. And as we shake them right and left, they basically spread out. So bees inside the swarm collectively move around in the swarm and they change the aspect ratio of that cone. And then when we stop applying this mechanical perturbation, then the bees again move around and they collectively go back into the steady state configuration. So I don't have time to go really into a lot of details, but we also connected that experimental result to a computational model where we treat the swarm as an elastic material. We have spherical bees that are connected by springs, so we can make some predictions about the local strains and stretching of bonds that an individual bee would experience. And we note that this could actually be used as a local mechanical signal to drive that behavior of the bees. So for elongated swarms, there's going to be more of that stretching of the bonds. It's actually going to occur upper in the swarm, so closer to the attachment surface. And when the bees move to a more spread out swarm, they effectively reduce those mechanical stretchings. And we can, of course, also reproduce this with an agent-based model. So our bees in the model can actually sense and respond to that perturbation. And just a funny anecdote, or I mean, I guess kind of a fun feature of that model is that when bees move upwards towards the base of the swarm, they actually move from areas where there's low amount of stretching of bonds to areas where there's a higher amount of stretching of bonds. So in a slightly more philosophical note, this is a little bit like mechanical altruism because they do this for the greater good of the entire swarm. Okay, so I showed you a steady state analysis of these materials, a more dynamic mechanical perturbation applied to these materials. But what about growth and how do these materials actually form? This is something that my lab is actively working on. We are interested in the communication signals that the bees use in order to create these swarms and these structures. The bees use basically chemical communication very much like cells do and social amoeba do in order to aggregate, but they use a trick where they actually direct the signal by creating airflow with their wings. So beyond the obvious advantage of using airflow, that is being that the signal is going to move faster, it also must have some kind of advantage in terms of the directionality that this airflow creates. And we studied this in the lab. So over here, you see a simplification of that process in 2 dimensions. We have the queen at the upper right side of the experiment. We started with the bees at the lower left side, and they were basically scenting. So every time you see one of these blue arrows, that's when a scenting event occurred. This, by the way, was done in collaboration with Greg Stephans, who helped us enormously by developing a very nice machine learning code in order to detect those in a high throughput way. So we can take those directional signals, we can kind of integrate them, and we can see that they indeed lead the bees to the queen. We can also use the combination of agent-based models to try to understand what is the benefit of that directional signal that the bees are using. And all of this is in 2D, and we are also interested, in what happens in 3D. So I'm just going to show this one movie here on the right side, where we are trying to understand how bees integrate signals that are chemical but also mechanical when they're forming those large aggregations. So these are the 3 examples that I wanted to show. Keeping these in mind, I think there's a lot of already ideas that are formed in the literature about active matter that are designed to explain active matter on a smaller length scale, on a cellular length scale, like what we heard today from Cristina about manipulation of the elasticity tensors. This is mainly the theory done by the Vitelli group in University of Chicago. And how do we take this and basically apply this to a larger length scale? As the title of my talk suggests, how can we think about growth and form on a larger length scale for larger organisms that maybe need to deal with a completely different set of environmental information or forces like gravity and so on, that becomes very important on this scale. So I want to finish with just a kind of a broad call and mention that I think there's a real opportunity here to think about creating this kind of theories for active materials on a larger length scale that also capture a very wide range of shapes of individuals. So far, I told you mostly about bees and ants that could somehow be, considered to be circular or very, not very wide ellipse. But there are also other aggregations of insects like these fungus gnat larvae, where juvenile basically traverse very rough terrains and are able to move as a big group. And of course, as soon as you start considering more elongated organisms, then it's going to be, you have to take into account entanglement and so on. And this becomes even more obvious for worms, for example, that are much longer and flexible. So there's a beautiful work that Saad Bamla and Dan Goldman for Georgia Tech are working on. I hope this movie plays. So these are worm blobs that are also able to navigate together as a group. They, of course, if you want to think about understanding the theory behind this, you have to consider that you're now working with polymers air brackets rather than colloid air brackets. So with that, I'm just going to thank my collaborators. Here's El Mahadovan again. He was my postdoc advisor, and Greg Stephans, and my new lab that I'm very excited about. Thank you for your attention. Sorry for all the technical difficulties. I hope I could get the message across, and I'm happy to take any questions.
Thank you for a very interesting talk. Let's see. So we have a question from the chat about the insect aggregates on the branches and other surfaces. Do you track the insect fraction in these aggregates? And if so, does that tell you anything about the system?
The insect fraction? Is that?
Do you track, like, let's say the density of the insects within?
Oh,
is it
right.
So do you track that, and does
very
that tell you anything? Do you have any sense there? Maybe another related question would be, how do you know what's driving their deformation when you perturb them, and what they're trying to maintain or optimize or anything like that?
Okay, so first of all, maybe your interpretation of the question, what we think is going on is that the bees try to minimize local deformation that would experience, with bonds that they make to their neighbors. So we think this is what's driving the shape-shifting behavior of that structure. But we're completely working under the assumption that the density is homogeneous inside the swarms. And until very recently, we could only see what's happening on the surface of the swarms. We don't really know what's going on inside, so we cannot really track the density there. But I think you can still see my screen, and so we're at the moment working on X-ray analysis of these swarms by basically performing a three-dimensional reconstruction of the density inside the swarm, and we can already see that our assumption breaks down when we work with very large swarms. So at higher locations in the swarm, closer to the attachment surface, the density seems to be higher in comparison to the tip of the swarm, which makes sense mechanically because the bees at the top of the swarm would be the load-bearing bees. So we're working on it. No concrete conclusion yet, but we're working on it.
Great. In the model you had for the bees, the balls and springs, and the agent-based models, it seems like you're modeling these as spheres. Do you have any comments on how the asymmetrical shapes of the bees may influence the overall behavior or other aspects of the mechanical response?
Right. So no, I don't. This is kind of related to what I tried to point out in my last slide, that we know our assumptions are too simple. We did them because we were not able to capture that complexity until now. But I think this is a very interesting direction to take that research to, both take the actual aspect ratio of the bees, which is of course not circular, but also push that to the limit where we have very, very long and flexible organisms.
Great. Let's see. Are there any other? Oh, Eleni has a question.
Yes. So it is maybe a question that maybe combines a little bit what you talked about and what Michael talked about earlier. So I know this is an extremely computationally expensive experiment to do, but would you think there would be any value if you try to approach this from an evolutionary algorithm perspective, right? So you have like a swarm where the behaviors of the bees are, the rules are maybe random, and then you do incremental adaptations, and you figure out whether the system can evolve this type of things, because it's beautiful. I mean, it's really puzzling how I don't know how smart bees are, but this is a collective behavior that it seems to me evolved out of, thin air.
Mm-hmm. Yes, I think that's a direction that actually some people take more for the sparse collective behavior that I mentioned. As far as I know, there's evolutionary algorithm applied there where you don't tell the agents anything about the behavior, and they're free to choose from those options. But I'm not sure I know any applications of that approach for more dense materials, and to try.
Thanks.
Right. Josh has a question.
Hey, Orit, I have an ill-formed question, I think. So when you think about, aggregated phases of of molecules or even cells, there's the idea that they really can't generate, or they don't have long-range information. I mean, maybe you could do it some fancy way, but you know, they can smell what's right next to them, but they can't see the whole thing. Animals, of course, can, right? Like if I'm in a bird flock or a fish school, maybe not inside the bee aggregate, but you know, you could just act as an individual and not, you know, and the group could achieve things without having really there be these sort of cohesive collective behaviors where, you know, individuality doesn't matter so much and things like that. So I guess my question is it clear when you want to evolve these kinds of collective mechanisms?
Sorry, so my internet connection is unstable and I could sort of hear your question. So is your question about using local information versus global information?
Well, I think there's this idea that you can make interesting population level changes with simple rules. Uh-oh. Can you hear me, Orit, or no?
I can hear you now.
Okay, so you can either have every agent do very simple things, and then the collective can do something cool, or every agent could just do very complicated things. And so is it clear when evolution sort of takes advantage of the more collective nature of things versus the individualistic nature of things?
Okay, so I think I understand the question. I would say that for this is something that the bees, I think, are kind of have this very impressive ability to work as a collective on very complex problems where they just don't have access to the information that they would need in order to solve that problem. So in a sense, it's almost like they managed a way to sense locally these very large physical fields of elasticity in the case of the swarm shaking, and know how to use that local information in order to achieve some global function that as an individual would be really, really hard to do. There are other examples of this for nest ventilation and for their aggregation process. And I think you also asked, if I understood correctly, about, you know, where is the limit? Where does it make sense to move from that distributed nature of sensing and solving a problem to, well, the answer that I don't know the answer to that. I think it's probably really depends on the problem, and I'm not aware of any universal ideas of how to address that, but it's very interesting.
All right, so we're out of time, but I want to thank all of the speakers for engaging few hours, and thank you to Tierra for helping put all this together, and Emory TMLS. I also want to thank National Science Foundation, Physics of Living Systems for support, and I don't know, Daniel, is there anything else that we should add at this point? Anything else you wanted to say?
No, just, you know, thank you to all of the speakers. This was really fantastic. And thank you everyone tuning in from YouTube for all these engaging questions and discussions that you've been submitting.
Thank you.
Okay. Thank you.
Thank you all.